அதெல்லாம் அப்புறம் பேசிக்கலாம். அவங்க இங்கே வந்ததுக்கப்புறம் இதை பேசுங்க. இதோட முடிச்சுக்கோங்க. இப்போ சொல்லிட்டீங்களா, விட்டுருங்க. அவங்க வந்ததுக்கப்புறம் இதை ஆரம்பிங்க. சரியா? எப்போவுமே ஆட்கள் 3 பேர்த்த கூப்பிடுவீங்களா, அந்த 3 பேரும் வாங்கன்னு சொல்கிறத நிறுத்திடணும். அவங்க வந்து இங்கே உட்காந்ததுக்கப்புறம் தான் அந்த செஷனை பற்றி பேசுங்க. பார்க்க வரலான்றதுக்காக கேட்டோம் மேம். வந்துட்டீங்கன்னா பார்க்க வரலாம். இல்லை உள்ளே அரேஞ்ச் பண்ணிட்டு இருந்தோம் மேம். சரிங்க மேம். ஓகேங்க மேம். இப்போ எதாவது ஸ்பீக்கர்ஸில் நோட் பண்ணிட்டு இருக்காங்க மேம். இன்னொரு 5 minutes ஆகும் மேம் ஸ்டார்ட் பண்ணுறதுக்கு. சரி. சரி ஓகேங்க மேம். உருண்டு கீழே வந்துருச்சு மேம். வந்துட்டு இருக்காங்க. அதுக்கப்புறம் வந்து இங்கே எல்லாமே பண்ணணும். இதுதான் ஃபஸ்ட்டு சொல்லிட்டு அப்புறம் தான் இது எல்லாமே வரும். அதுதான் நான் என்ன சொல்லுவேன்னா, அதாவது இன்ட்ரடியூஸ் பண்ணுங்கன்னு சொல்லும் போதே டு வெல்கம் அண்ட் இன்ட்ரடியூஸ் தி ஸ்பீக்கர் அப்படின்னு போட நிறுத்திடணும். அப்போ இவங்க சொல்லிட்டு வெல்கம்னு அவங்க சொல்லும் போதே அவங்க போயிடணும். இது நீ அமைச்சு போய் நின்று கூட ஆமாம். இங்கே திரும்பி திரும்பி பார்க்காம இருக்கும். அது ஒன்று தான். நீ யாருக்கு கொடுக்கணும்னா ஹெச்ஓடி இருப்பாரு இல்லையா, ஹெச்ஓடி யார் இருக்காங்களோ அவங்க கிட்ட கொடுக்கணும். அந்த பேர் போயிருக்கோம். அங்கேருந்து ஐடிக்கு இது இருக்குது, ப்ளசன்ட்ரி இருக்குது. அப்புறம் எப்படி சும்மா தான் கொடுக்குறது? படிக்க ஒன்றுமே வெள்ளாமல் விடுற இல்லைம்மா. அவனுக்கு இருக்கணும். இவருக்கு
அப்புறம் எங்களை கூப்பிடுங்க. அவசரப்படாதீங்க, பார்த்துட்டு கூப்பிடுங்க. அப்புறம் பார்த்துட்டு வந்து பேர் நான் ஸ்டார்ட் பண்ணேன். ஸ்டார்ட் பண்ணலாமல் கேள்வி ஸ்டார்ட் பண்ணேன். வந்தோடனே ஸ்டார்ட் பண்ணிடுறாங்க. சரி பரவாயில்ல. இப்போ வந்து இந்த போஸ்ட்டை பற்றி அவரே சொல்ல சொன்னார். போஸ்ட்டை பற்றி சொல்லிடுங்க. அதெல்லாம் போடுறோம், போட்டிங்க வந்துடுங்க. இந்த செஷன் பாருங்கள். போஸ்ட்டு போடுற மட்டும்தான் போடணும். அப்படி தான் போக மாட்டான். போஸ்ட்டு போடுற மட்டும்தான் போகணும். சரி வாங்க. சரி 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 அப்புறமேல் பண்ணிங்க. அப்புறம் சொல்லி நிறுத்துடா. அவங்க வந்து உட்காந்ததுக்கப்புறமா ஆரம்பிங்க. செஷன் முடிக்க என்ன பண்ணுங்க? அந்த போஸ்ட்டை பற்றி அதாங்க சார் பேசும்போது நீங்கள் சத்தமாக பேசுங்க. போஸ்ட்
We welcome Dr. Pariyapon, Assistant Professor and Head from Maharasarakam University, Thailand, to come to the dais. We cordially invite Dr. Chang Woo-ji, Research Professor from Yesu, South Korea, to also join the dais. We request Dr. B. Ramesh, Associate Professor, Department of Biochemistry, to join this session.
ஃபாஸ்ட் பண்ணுங்க. அவன் குழந்தை கொடுத்து
which keys you wish to windows எடுத்துக்கிறாரு அதான்றீங்களா அவ்வளோதான் அவ்வளோதான் சூப்பர் சூப்பர்
இது க்ளோஸ் பண்ணி விட்டுருங்க. ஓ ஐ சி.
இன்ட்ரடியூஸ் பண்ணுறது
எஸ். Can yeah. It didn't come actually. Change of button. Slide change of it is different one.
One second, be seated. You might make it ready. You can move with this.
Yeah, sure.
This is forward.
Yeah, I know. Yeah.
I'll get it ready now in 2 minutes.
Okay, no problem. Okay.
Check.
Good morning to all to the second day session of International Conference. I am very glad and consider it a privilege and an honor to introduce the Arthur Hoyser Awardee, Assistant Professor Pariyappan Isranwad, who graduated with honors in food technology and nutrition from Mahasarkam University, Thailand. She got PhD in food and biotechnology from University of Reading, UK in 2003 under the Royal Thai Government Scholarship. Before coming back to work as a lecturer in the Department of Biotechnology, Faculty of Technology, Mahasarkam University. Pariyappan received a number of awards and scholarships such as the Arthur Hoyser Award from the University of Reading, the Don Whitley Travel Scholarship from the University of Reading, the second FOSS Prize for the Dairy Science from the Society of Dairy Technology, the President's Fund from the Society for Applied Microbiology, JSPS Scholarship, University Mobility in the Asia and the Pacific UMAP Projects Scholarship, the Young Scientist Meeting Grant Award from the Federation of European Microbiological Societies, the ASEA UNI NET Scientist Exchange Scholarship, ASEM DUO Fellowship Program, and Invited Professor Fellowship at École Nationale Supérieure Agronomique de Toulouse, France. As an active scientist, she has been conducting many research projects, both national and international, and collaborating with several countries such as UK, France, Austria, Japan, UAE, Singapore, Malaysia, and India. Her expertise is in the areas of probiotics, prebiotics, lactic acid bacteria, nutraceutical and functional food, food biotechnology, food microbiology, herbs, and cosmetics. Pariyappan is very outstanding and successful in bringing research into industrial sections. She serves as a consultant for more than 500 small and medium-scale enterprises and community enterprises all over the country under many projects supported by the Department of Industrial Promotion, Ministry of Industry, Office of Small and Medium Enterprise Promotion, Community Development Department, Mahasarkam Provincial Cooperative Office, and Royat Cultural Office. Furthermore, she hosted the annual International Training Course 2003 entitled Value Added and Quality Improvement of Agricultural Products by Fermentation Technology for 84 participants from 24 different countries, supported by Thailand International Cooperation Agency, Ministry of Foreign Affairs of Kingdom of Thailand. I request Madam Pariyappan to deliver the keynote address on the topic Lactic Acid Bacteria, the Promising Cultures for Bio-Business and Better Quality of Life from Research to Market. Madam, we are all ears for you. Welcome.
Vanakkam, Sawadee and good morning. Okay, so how are you today? Fine, you are fine. Are you ready for today? Yes, very good. I just apologize that when I changed to I mean sometimes for the new system and the font is come different. Yeah, but it's okay. First of all, I would like to thanks to the organizing committee for your kind invitation to share my experience in this international conference. It is always my great pleasure to be in Coimbatore. And today I would like to share my work about lactic acid bacteria. Yeah, for the lactic acid bacteria, I'm not sure if some of you are familiar with this bacteria. You familiar with this one? Yes? What is the product that we use lactic acid bacteria for fermentation? Very good, yeah, for the curd. And also for some fermented food, some fermented vegetables, fermented milk, and also for some fermented meat. And for my work, I work on food biotechnology, functional food, and fermentation. for the lactic acid bacteria, usually lactic acid bacteria is gram-positive and gram-positive and catalase negative. And for this one, they have different shape. For example, they can come in cocci like this one that you see, okay? And in okay. And this, yeah, we just wait for this for some time. Okay, no problem. Okay, here we are again. Yeah, for the lactic acid bacteria, sometimes it can be in cocci like this one for the streptococcus and what else? Yeah, the enterococcus. Someone here have a background in microbiology? Anyone here? Yes. So for this one, you know, for the bacteria, they have different shape. For example, cocci. And next one, please. Yeah, and sometimes in rod. For example, lactobacilli. Next, please. Yeah, and for this one, usually for the lactic acid bacteria, means they can produce lactic acid from using kind of sugar. They turn from sugar to lactic acid. We can classify lactic acid bacteria into two groups. The first one is homofermentative group. And for the homofermentative group, this one mainly produce only lactic acid. And for the second group, this one we call for the heterofermentative group. What is the hetero? Hetero means more than one, right? And the heterofermentative group produce not only lactic acid, but it can also produce for lactic acid, acetic acid, and ethanol and carbon dioxide. Yeah, and next please. What can we use lactic acid bacteria for? We can use it as starter culture, for example, in meat product, in vegetables. I think in the middle picture, Dr. Chi might be familiar with that. Kimchi, yeah, right? Dr. Chi, is it kimchi? Yeah, it is kimchi. So for the kimchi, we use lactic acid bacteria as the dominant microorganism for fermentation. And also we use it for dairy products and some meat. Next, please. Yeah, let us see what are criteria if we would like to use lactic acid bacteria as starter culture. The criteria for culture selection are, yes, please. Yeah, next one, yeah. We see for the production characteristic of the strain. First one we see for cell preparation. Cell preparation for determination of biomass production. What does this mean? Usually when we produce starter culture, What do you want from that? Of course, we need biomass. We need a lot of cells. So for this one, we have to check if they can produce a lot of the biomass. So for this one, we see for the cell preparation. In terms of cell preparation, what you have to look for? You look for the media. Is the media is expensive or cheap? Sometimes it's good, it gives high yield, but it's not practical. You cannot apply it because the cost is too expensive. So for this one, you see for the cell preparation, the condition of fermentation, and also media that we use. And second one, we see for the yield after fermentation. Of course, when we want to produce something, the thing that we want is high yield. I used to discuss with my colleague. She researched in some bioactive compound, very interesting. And then I asked, what is the yield? Only 2%. In terms of commercialization, 2% is too small amount. So for that, we have to think about if we would like to do research and move it forward for the commercial scale. The thing that we have to think, about is the yield of production, and also the separation of biomass. Usually, to separate biomass, there are different techniques. If that living thing is big, have the big cell, it's easy. You can easily just filter it. But for microorganism, the size is very small. Normally, how can you separate for the biomass? Can anybody of you share with me here? For example, we prepare microorganism in media, let's say in broth, and then we would like to collect just only the cell or the biomass. What is the technique that you use for separation? Centrifugation. Yeah. We can use centrifugation, we can use filtration, but filtration for bacteria maybe is not that practical. So for this one, we have to see the appropriate technique to separate the cell. And next, for the stability after lyophilization culture. In order to use the culture, usually we keep culture in dry form. And to keep it in dry form, normally the technique that we use is feed drying or lyophilization. Okay? This one that you have to pay attention. Next one, please. Yeah. And also, for the technological characteristic of strain. Technological characteristic of strain means the property of strain that they will give you as you like. For example, if you want to produce acid, the strain should be able to produce acid. So for this one, we check for acidification activity. Of course, lactic acid bacteria, the end product should be lactic acid. So for this one, we check for that. For if it's capable to produce lactic acid as we want, because some strain, at the beginning, the strain is strong. It can produce like you as it is or as you want, but after you keep it for long time, it have mutant, it kind of some mutation. So for the mutation, you have to see also the ability of the strain. And see for the aminopeptidase activity, what is aminopeptidase activity? This activity is necessary when you would like to use starter culture for dairy products and even for meat product, because dairy and meat contain protein. So we want bacteria to produce to produce enzyme to digest protein into amino acid. At the end, after digestion, break down the molecule of amino, it improves the flavor of the product, and it also improves for the sensory evaluation, the taste, the smell, and also texture. And we see for the autolytic activity, flavor development, and antagonistic activity. What is antagonistic activity? Do you familiar with this word? Antagonistic activity means two things that against each other. So for this one, we check for the antimicrobial activity. For example, we have lactic acid bacteria that is good bacteria, and we want to apply it in product that might contaminate with some pathogenic bacteria or even some spoilage bacteria. So we check if they can produce some metabolite, for example, organic acid and some other metabolite that can inhibit, okay? And next one for the exopolysaccharide production. This one help to improve the texture of the product. Next, please. Yeah, and let us move to the word probiotic. Do you know what is probiotic? Nowadays, it is very trendy. In functional food area. So for the probiotic, let us see for the definition of these three words. When you see the word probiotic, you will see for prebiotic and also for symbiotic. Just briefly, probiotic mean bacteria or yeast, mean microorganism that is good for our health. And prebiotic means substances that stimulate the growth of probiotic. And symbiotic means the combination between pro and pre together. Okay? So now you know what is probiotic, prebiotic, and symbiotic. Yeah. And for this one, like I said, to be considered as probiotic, the strain should be able to survive through the upper part of the intestine. So it go past stomach, small intestine, and go down to the colon. Yeah. So for this one, if some strain cannot survive through the upper part of the intestinal tract, it is not probiotic. For the probiotic, when it reaches colon, it colonizes itself and use prebiotic and produce some metabolite, so-called short-chain fatty acid or some organic acid, and stimulate the immune system and provide some health benefit. Okay? So see, and in healthy food industry, now they tend to use probiotic bacteria in combination with the original starter culture. They can combine together or just use separately. Yeah. And for this one, the probiotic health benefit, there are many benefits that they claim. Sometimes it boosts the immune system and is helpful for the allergy, for weight loss, and also to keep balance for the microbiota in our body, to keep balance between good and bad bacteria. You know, our stomach is our second brain. If your stomach working well, so your body will function well too. So just make sure that you eat well, you have good condition for your stomach, because our stomach is the second brain. So for this one, if we have balance of the microbiota, so means your body function well. And for this, it can also improve with somebody who have problem with diarrhea. And for the mental health, a friend of mine, actually she's Malaysian. She got her PhD from Australia, and she worked with probiotic. And I mean, for the function of mental, she fed mouse with probiotic. And then just to see the behavior from the research suggested that the probiotic that received probiotic, the mouse that received or got probiotic tend to stay calm. So for that one, just to prove that it can improve for the mental health. Okay, next one, please. Yeah, and this one I just would like to show you for some benefit of probiotics. For example, it reduces the risk of cancer. It can help to reduce cholesterol because when we take some probiotic can produce bisulfite hydrolase enzyme, and this one it can turn the cholesterol into deconjugated cholesterol, and then it can get rid of our body. So as a result, cholesterol reduce, and it can improve some immune system too. There are different benefits. Yeah, and yeah, let us see for the criteria. If we would like to select the strain, I think some of you are microbiologists, and some of you may be interested in working on bacteria or probiotic. And what are criteria for that? If we want to use the strain, what would it be important? The first one, if we would like to use it for human, it should be from the human origin, or sometimes from fermented food is okay because it's safe. And safe for food and clinical study. Survive during the gastric transit. To survive past to the gastric juice, for example, for the acid and bistability. Acid, what is the, where? Where is the acidic condition in our body? I mean for the, stomach. What is the range of pH in stomach? Between 2 to 4, depending on what inside your stomach at that time. So for the probiotic, it should be able to survive in acidic condition, and also to survive for the bisalt condition. Bisalt, we find it in small intestine, right? Yeah, and for this one, pH is about 8. So it can be able to survive and go to the colon and colonize itself. So when you study, just make sure that you study for the survivability during the tested condition, acidic condition, bisalt condition, and even sometimes we study for the survivability in the simulated gastric juice. You can decide. And to see the adhesion behavior to the gut epithelium tissue, and test for the clinical study. And of course, to apply, it should have good technology property. Good technology property means it should be able to survive well. Usually for the probiotic, they are very sensitive. I don't understand them. Very sensitive. I have to look after them very well and check the survivability. Some strains are good, but very sensitive. Can we deal with that? Anybody here are food technologists? Yes, some of you are food technologists. Is the work for food technologists? Because for microbiologists, we already got the good strain. So for the food technologists, you have to find the way, technology to preserve the cell because we get good one. We want to use this one. Usually for the technique that we use to preserve cell, we use encapsulation. You can use nano encapsulation, micro encapsulation as you like. One of my PhD students from the UAE, United Arab Emirates, she worked on nano encapsulation of Bifidobacterium lacteus, to prolong the survivability. And next one, please. So for this one, we check also for the health promoting property, such as if it can stimulate the immune system, reduce cholesterol, and to prevent some cancer or tumor. And some strain have function for the detoxification of the toxin. I have worked with the French Institute. One of my PhD students. We have the strain that we isolated from human colostrum. Interesting, right? From human colostrum. And then we check for the probiotic property, and we check for the detoxification of toxin. We got very good result. And this one already published. Next one, please. And so let us see the way to deliver probiotic. How can we deliver probiotic? The first way, by taking fermented food. This one simple. You just have your raw material. For example, for curd, raw material is milk. So we have milk, we have microorganism, you ferment it, and then you got probiotic products, and just eat it, easy. This one the first way. And the second one, to be considered as probiotic products, it should have sufficient numbers. The minimum number that required for the FDA is at least 10 to the 6 CFU per gram, okay? So when you claim probiotic product, you have to make sure that at the end of the shelf life, not only when you produce, end of shelf life, the number of probiotic bacteria should be higher than 10 to the 6 CFU per gram. And for the other way is you can have it by via the non-fermented food, because somebody may not like sour taste, right? Anybody of you here doesn't like to take something sour? Okay, so you can, it's okay for you to have some fermented food, but somebody, they don't want sour food. For this one, we can prepare them separately. First, we prepare just only for the cell of probiotic by using media, the suitable media. Anybody here working on lactic acid bacteria? What is the suitable media for the lactic acid bacteria? of lactobacilli strain in human and animal health is not only in food, but also important for feed industry, for the probiotic. Okay, you see that they use the Lactobacillus rhamnosus, the Lactobacillus reuteri in different kind of product. Yeah. And this one, the other genus is called Bifidobacterium. Bifidobacteria or Bifidobacterium, these are very sensitive. They are sensitive to oxygen. Usually, they are obligated anaerobic bacteria. Only one strain, it called Bifidobacterium lactease, that can tolerance to 10% of oxygen. Before, it used to be kind of synonym, if we study on taxonomy, is synonym of the Bifidobacterium animalis. Yeah, it's very similar to each other. The similarity is very close, okay? So for this one, it can also inhibit the growth of pathogenic bacteria, such as E. coli, Helicobacter, and also to support the immune system. Yeah, it have anti-toxic action also. Next, please. And for this one, there are different strains that been used in industry. For example, the Bifidobacterium infantis or Bifidobacterium longum. When you go to the supermarket, you can check on the dairy section and see, just curious, what they use. Usually, when I go to the supermarket, I like to see for the product. Even when I came to India, I went to the shop and see what they have in the market. It's very interesting to see product in each country, to have an idea how to develop the product. Okay, next one, please. Yeah, and these are some benefits of Bifidobacterium. For example, they can produce some vitamin and improve for someone who have lactose intolerance. At least one person here has problem in lactose intolerance, and that person is a chairman. I just worry that he will feel asleep, so I just mentioned. Dr. Chi? Yes, yeah. Dr. Chi has problem with lactose intolerance. Yeah, even in Thailand, because we don't consume that much of dairy product. So when you take dairy product, we don't have lactase to digest. So for this one, when we take fermented milk, fermented with probiotic strain, is already changed from lactose to lactic acid. Therefore, it can improve the problem of people who have lactose intolerance, okay? And reduce the serum cholesterol, prevention of diarrhea, and prevention of cancer. Yeah, next, please. And the other strain is Lactococcus lactis. This is my strain. Like I said, that I have been working with lactic acid bacteria and probiotic for many years. And in the lab, I did isolation. Actually, it's not just only Lactococcus lactis. We got also Lactobacillus. But why I decided to use this strain for upscale for the commercialization? Because it's already in the list of the GRAS. Do you know GRAS? G-R-A-S. Generally recognized as safe. So this list is from the US FDA. When you would like to use any kind of substances, microorganism in food, you would like to use food ingredients, microorganism, the most important thing is you have to check if it's safe to be used. And even for the microorganism, we have to see if this strain is safe. And for the Lactococcus lactis, it's already in the list, so you don't have to prove anything. You can just go for that. But some strain, for example, my strain, the other strain that I isolated from human colostrum, they have very good property for the probiotic. But the problem, can you go back please? Okay, yeah, so yeah, we stay here for a few seconds. Yeah, and for the Lactobacillus pentosus that I isolated, this one is not yet in the list. So if we would like to commercialize it, we have to study, have to prove it is safe. To do that, we have to study from the in clinical study to see for the safety and toxicity, everything. When it's proved, so we can use it in the product for commercialization. But for the Lactococcus lactis, it's already in the list. You can just directly use it. Yeah. So for this one, as I said that, it has been used to prevent some pathogenic bacteria because the property of this strain, they can produce some antimicrobial substances such as organic acid. But one good thing for this strain, they produce bacteriocin. Do you know bacteriocin? Do you know penicillin? Penicillin is antibiotic, but bacteriocin is the substance that produced by bacteria and have the ability to inhibit pathogens. But it's safer than antibiotic because mode of action are different, okay? Yeah, so for this, the other strain that we use is like Streptococcus thermophilus. Yeah. And Streptococcus enterococcus, and next one please. Yeah, and the other strain that now they tend to use it in human products and also for animal feed. For the animal feed, they use this bacillus for long time. The good thing about lactobacillus, about bacillus, only bacillus, they can produce spore. So when they produce spore, it means they are very strong, very tough. So it can have the high survivability rate. So for this, at the moment the trend, before we don't use it for food, only for feed. But now it is already applied in many food products, okay? Because it produces some spore, so it have the high survival rate. Next, please. Yeah, and even for the Saccharomyces, Saccharomyces can be used as probiotic too. I think maybe some of you know the product called kombucha. You know kombucha? Yes, you try it? No? Yeah, for the kombucha, it's also probiotic. But the difference between other probiotic is these have combination of lactic acid bacteria and yeast. So the product contains not only for organic acid, but can be also some carbon dioxide, okay? A little bit of carbon dioxide and a little bit of alcohol. So if you would like to try, I think they sell it in the market, yeah. And so these are some beneficial probiotic strains. If you would like to use some of these, yeah, you see and you check for the the property that they provide. For example, the Lactobacillus casei support the immune system and inhibit Helicobacter pylori and help to fight with infection. And other strain you can check. Yeah, and for this one, I just summarize different kind of product already in the market. You see that they use different strains of probiotic, and sometimes it's dairy free or not. If it's dairy free, it means with somebody who has problem, they can take it. And also, some product need to keep in the fridge because it's in because it's like in curd or in yogurt form. But if it's in dry form, you don't have to keep it in the fridge. For example, in capsule. Okay, next please. Yeah, so these are the best probiotic strain. If you would like to use in specific purposes, you go and you see. If you want to use it as the product to stimulate the immune system, you pick one. And if you want to use it for somebody who have problem with allergy, you check and what is the strain should be used. Yeah, and these are pro and prebiotic. Prebiotic can be found in different kind of vegetable and fruit like this. And even for probiotic, at the moment, they add it in different product like this. Yeah. Yes. Yeah, and the prebiotic I have already mentioned is substances that, substances that shouldn't be neither digested or absorbed at the upper part of the intestinal tract. So it should go and reach to the colon and stimulate the growth of probiotic bacteria. These are some prebiotics such as fructooligosaccharides, lactulose, what else? Xylooligosaccharides. They are different. If you go to the market, you might see they put some fructooligosaccharides, some inulin in the product. Yeah. And for the prebiotic, when we add together with probiotic can use and stimulate the growth and produce some short-chain fatty acid that provide health benefit to the host. Yeah, and symbiotic is the combination between pro and pre. We add two together to make sure that you will not miss anything. You have both pro and pre. So it calls syn. It's not symbiotic, okay? Different. Symbiotic is the support of two living things to promote the growth of each other. But symbiotic is combination of pro and pre together. Yeah, and this one I just to summarize for the probiotic and prebiotic. They should go and survive up to the colon. And even for the prebiotic, shouldn't be digested or absorbed on the upper part and reach the colon. Yeah. And this one I just talk about it briefly. When we use lactic acid bacteria, sometimes we can use it as a source of biopreservative because it can produce so-called bacteriosin, and this substance can prevent the growth of pathogen. Yeah. And for this one, usually for the perishable food, when we apply fermentation technology, we can extend shelf life. Why we can extend shelf life? Because lactic acid bacteria can produce lactic acid and other end products. Other end products such as hydrogen peroxide, diacetyl acetylene, and other substances such as bacteriosin, and they can preserve the food. Yeah. And for this one, many strains of gram-positive bacteria can produce and we can apply because it is recognized as safe, and the major the majority of bacteriocin producing strain are lactic acid. Okay, lactic acid bacteria, yeah. And these are niacin that already used for commercial strain, commercial food ingredients. Next one, please. Yeah, so we can use bacteriocin to improve food safety. To use, there are 3 ways. Yeah, for this one, we can use the strain that produce bacteriocin, and we purify bacteriocin and then apply it in the product that you like. Or the other thing, you can purify or semi-purify and use bacteriocin culture. For this one, we call in situ production. You use the strain that produce bacteriocin together with starter culture and let them grow. During growing, they produce bacteriocin and preserve the food. Yeah. And for this one, sometimes if you work on purification of bacteriocin, this one can incorporate with the packaging technology. We can produce bioactive packaging, especially the one that contact with food, by add some bacteriocin in the materials, and then you allow them to release. You have to study for that, okay? To allow them to release on the food. So for this one, it can prevent for food spoilage. Yeah. Yeah. And this just only some influence factor that affect to the ability of bacteriocin. For example, different strain, the metric of food, the composition of food, and these are some factors. Next, please. Okay, so now we move to the last part. I just take the time to, okay. And how can we turn from research to market? I just give you the not case study, but some example that I apply from my research to market. This one produced by the company called Cost Bioinnovation. Actually, I found that I established this company maybe 5 years back. And this company have a purpose just to bring research to market. Yeah, it's my dream, you know. As a researcher, the most important thing when you research, you should be able to use it, right? You research and it will be used in public. So for this one, I work hard for that. I study for lactic acid bacteria, and not only that, like I was introduced at the beginning, I work as a consultant for many factories, and not only companies, sometimes for the SME, the small enterprise, everyone. I just love to do that. It's my passion to develop the product and to see it in the market. Sometimes many SME just call me, and it's my pleasure just to help them, okay? But this one is for my project, yeah. So for this one, we successfully isolated and studied well for the Lactococcus lacteus. The strain is A7. So for this one, actually I have to show you. This one is the product. I bring it from Thailand. Yeah, so for this, we start from isolation of lactic acid bacteria. The source of isolation is from traditional fermented Thai food. Okay, the traditional fermented food. And for that time, we use different kind of sources. For example, from fermented meat, fermented fish, fermented vegetables, and then we did isolation and test for the the gram staining, the catalase test, just to make sure that there are lactic acid bacteria. And then we study for the antimicrobial activity, the production of bactericin, because we are looking for the strain that can produce bactericin. Actually, this one we used to work with the French Institute to find the bactericin producing strain to be applied in food. Yeah, and for this one, we check for the property of the bactericin that we produce, and what else? Yeah, to see the stability in different pH. From this slide, I just would like to point out, in order to use the strain for commercials, of course, you would like to use it in different kind of product, right? You would like to apply it in juice. Usually the pH of juice is acidic. And sometimes you would like to add it in just milk, but not fermented milk. And pH is about 6.8. For the neutron, no problem. But sometimes when it's acidic or alkaline, we have to make sure that the property of bactericin is still there. So we check for the property for that too, to prove that it can be used in the very wide range of pH. And then we test for antimicrobial activity for the gram positive and gram negative of pathogenic bacteria. And then to see the production and mode of action. Yeah. And then we identify the strain, and it is identified as Lactococcus lactis, subspecies lactis. And fortunately, this strain already in the list. So means it's safe to be used. We don't have to prove anything else. But the strain that I isolated from different sources, sometimes it's not in the list. If I want to apply in food, I have to prove it. So that one is another project. Okay, next please. And then I test for the probiotic property, such as acid tolerance and other conditions. Yeah, and to test for the antibiotic sensitivity, yeah, like this. Yeah, and also when we check everything, we move to see how to use it as starter culture. So for this, we study in lab scale. First, we study for the production of lactase milk. I call it lactase milk because it produces from Lactococcus lactis. So we see for the incubation time, the pH, percent acidity, and viscosity. This just to find the condition for production. Yeah, and then, yeah, this just only for the step. First, we did for isolation, incubation, then feed dry. And at the end, we got the product in freeze dry form and then formulated. Yeah. Yeah, and this project under supported by the National Innovation Agency of Thailand. Yeah, so for this, for the project, I have to deal with 2 factories. For me, my way to do a business, sometimes you don't have to own your factory. I'm too busy. I don't want to take care of everything. So how can I do that? We just do OEM. So it means you ask some factory to do it and you pay them. So for this one, it's good for the beginner because you don't know yet if this product is okay for the market or not. So for my way, have some other thing to do, and the way to control it easily, we select the factory that is qualified. For example, I use the milk, the dairy product factory to produce my lactase milk, and this factory should have already certified by GMP, GSP, HACCP, and everything, okay? So we upscale for commercial scale there by fermentation and validate the process. And then the factory 2 is the second step for feed drying process for lactase milk powder. So we, actually this one is the feed dry. So we, after production of lactase milk as a curd, we transfer it to the second factory. And at this factory, we got the powder, like this one, okay? Like this powder. And when we get this one, I expected it to get approved for the FDA, I mean, to get the FDA registration number by these 2 months. Actually, this month and next month it should be done, okay? When I get the product, I share you the information. So for this, we just formulate and then test for the property that we have to apply to get approved by FDA. Yeah, and that is one product. And this one is the, we are on time. Is the one of my students' master student work. In my department is biotechnology. I have background in food technology and nutrition. And then I study for PhD in food biotechnology, so my expertise is on food biotechnology, functional food, and so on. So for this, as a head, I modify a little bit for the curriculum because I have passion of bringing research to the market. And even I like being a business girl from I was a little child, very young, I earned money when I was very young. So I just want to stimulate, sometimes not stimulate, but to share my idea to students. So for our curriculum for bachelor degree, master degree, and PhD, we combine biotechnology together with entrepreneurship. So for the bachelor degree is in biotechnology and bioindustry. For the master degree is biotechnology and biobusiness. And for PhD is innovation in biotechnology for business. So for this, we teach how to use biotechnology to add value or to solve problem. For example, we have also for wastewater treatment in one section, okay, and agricultural technology, biotechnology, food biotechnology, animal biotechnology, and environmental biotechnology for that. And we teach students to be an entrepreneur, to have their own brand. And this, I just would like to show you one of my students for the success case. She is Mrs. Kanya Park, and at the moment she have the company called Kanya Thai Cuisine. Actually, she is just only the first year student for master. She enrolled for the master degree for biotechnology and biobusiness. And now she have her own product and already sold in the market For example, for the tempeh, I don't know if you know tempeh. Tempeh is the soybean product. Yeah. Fermented with what kind of microorganism? Fungi or mold. Is Lysopus oligosporus. So for this one, after fermentation, the fungi or the Lysopus oligosporus produce some enzyme, and then it can hydrolyze protein into amino acid and some other metabolite that can improve health. Okay, so for this one, she have a product in freeze in the raw tempeh and in freeze dry tempeh, and recently she just got the product we call plant-based coffee. Plant-based coffee means usually people have coffee, just coffee with some sugar or whatever. But with this coffee, we add some tempeh as a source of protein, so that when you have coffee, it can also give you some health benefit to add some protein intake, especially for the one who doesn't have time, just drink coffee and go. So you just have sugar, have caffeine, but no protein. So for this one, we just want to answer the pain point of customer. You have everything in one, okay? So it's already in the market. Yes, next please. Yeah, so to conclude, lactic acid bacteria play important roles for the bio-business and human being. Yeah, and before I finish my talk, I would like to invite you to join the International Conference on Biodiversity, Science and Technology, which will be organized by my faculty. We are thinking to organize it in Pattaya, that is the seaside. It will be in 2024. Yeah, if you are interested, you kindly please answer the form. Yeah, for this one, it will come back. Yeah, so for the next one, please. Okay, thank you very much for your attention, and I hope you learn something from my experience. Thank you very much again. Sorry.
Please ask some question, Professor Oli. Is there any question? So I have one question, Professor Oli. Yeah. I don't know the meaning of how many species or genera LAP, lactic acid virus, in the world. How many lactic acid virus are known in the world? How many?
Yeah, actually there are about 13 genus, but there are different kind of species. So when we study on lactic acid bacteria, even though sometimes you have the similar genus, but we still have to study in details because each strain have different property. Yeah. So for this one, if you would like to isolate, yeah, first, I mean, they are different. There are many strains for that, depending on the source that you do isolation. And then when you get the strain and the strain, you can easily identify. If you would like to do it, just quick identification, you can do for the biochem test. Yeah, like I show you. From that one, I check for the from the biochem test. And the other thing, to be more precise, we can do for the genetic test. Okay, thank you.
Excuse me.
Yes?
You told many bacteria in the milk. How do they behave inside the milk? Are they friends or enemies or stranger inside the milk? That is lactobacillus, bifidobacteria and other bacteria. Do you have any idea, ma'am? Can you get the question, ma'am?
You mean how can we identify the different?
No.
Sorry, because it has some echo. Yeah.
What is the interaction between these bacteria inside the milk? Do you have any idea, ma'am?
Yeah.
Ma'am, he's asking, you have done all the in vitro studies, isolating this bacteria and testing it on the plate and its biochemical characterization and all those things. He's asking how these bacteria will behave when it is present in the milk.
You mean, thank you. Thank you, sir. Thank you. So you mean when we apply it in milk, how they behave?
Yeah.
Okay. So for that one, even though we isolated from different source, from fermented meat or from fermented vegetables, the strain already there. It will be additive, but the behavior will be like when we apply it in different environment, we have to check for the nutrients. For example, in milk, if for milk there is no problem because milk is very rich in nutrients. So when you apply it in milk, bacteria grow as we study in MRS broth. But just only what we have to check, when we want to apply, we have to see first for the media or food that we are going to use it as substrate. And the second one is the condition of growing. The condition of growing, for example, if you are going to grow them in 30 degrees C or 37 or 42, there is things, if we grow them in ambient temperature, there is a matter of saving cost. Because sometimes if we have to generate the temperature up to 42 degrees, so means you increase the electricity bill, right? So for this one, to study, we have to check if we can grow them in ambient temp, or it have to be in the specific temperature. The behavior are different. Of course, if we grow them in optimum temperature, that can go fast. But if we grow them in not maximum, but they can grow, they might go slowly. But if it's in acceptable level, we can go for that, just to reduce the cost of production. Yeah, thank you. Is it clear, or am I answering your question?
Ma'am, next question, ma'am. Can I ask next question, ma'am?
Yes.
Ma'am, if you isolate completely a bacteria from a milk and grow in a lab, and then can you add same bacteria in the milk? Can it grow? And what will the quality of milk be?
Yeah, you can grow them. Yeah, we just have to set the condition as a suitable condition.
What will be the quality of the milk?
Sorry.
What will be the quality of the milk after adding the bacteria grown in the lab?
After adding bacteria.
Yeah, that is grown in the lab. It will affect the quality of the milk.
Mm-hmm.
It will affect the quality of milk or not?
Actually, you mean for the quality?
Isolate a bacteria completely from the milk and grow it in the lab. Yes. Culture it.
Yes.
And again add the same bacteria in the milk.
In the milk.
Milk. What will the quality of the milk?
It can work perfectly, no problem. Even though you do in the lab and then you add in the milk, because they are the same microorganism, we just have to control the condition. That is the most important thing. And also for the property of raw materials. But if you use in milk, it's still milk. So no problem.
That's why I think in the milk, it is in natural condition, it grow, it not affect the quality of the milk. But in the lab condition, we add many chemicals, the bacteria go in that condition, and then they add to the milk.
Yes, you can use that.
Okay.
Because for that one, you can use that fermented milk that you already added in as starter culture. You see what I mean?
Okay.
Yeah, you have lactic acid bacteria, you add it into milk, and you grow them. And then you have fermented milk. You can add this fermented milk into the other fermented milk, into the other new milk. Okay. So for this, we call this one as starter culture. You can use it like that. But just be careful for the contamination. Because if we use in household, you have to make sure that every step is aseptic technique. Okay?
Thank you.
Thank you for your question.
We spend too much time. If you have a question, you can ask to Dr. Professor Oli when break time. So let's finish this session. Thank you all.
Thank you, sir. Thank you, ma'am, for your interactive session on the importance of lactic acid bacteria towards enhanced metabolism and improved food safety that can aid in promoting overall health. Besides, it was an interesting session on sharing your research cum product development that would be motivated for the students to think on food-based startup as well. We are glad to invite Dr. Shanmugapriya, Professor, Department of Microbiology, to express our gratitude to the speaker, Dr. Pariyapon, along with the session chair, Dr. Chang Uji, through pleasantries. Thank you, sir. Thank you, ma'am. We extend our thankfulness to the speaker, chairperson, and co-chair for engaging this technical session.
Now, as we move on to the technical session 2, we are glad to invite Dr. Seraladan Kamalakannan, Dr. Pariyapon, and Dr. J. Sendil Kumar to please come up to the dais. Meanwhile, we have a small announcement to all the poster presenters. All those who wish to display their posters scheduled for today, as well as those who have missed the session yesterday, may please mount their posters at Brahmam Hall during the tea break. Please note it is during the tea break, and please return to the auditorium for the upcoming sessions. We will be notifying you when the evaluation starts. The same is applicable to the oral presenters as well. Those who have missed to present yesterday also may proceed to the respective venues at the respective time in order to present your work. Once again, please note, both the oral and poster presenters may proceed to the respective venue only during the tea break, and please join the technical sessions. Thank you. We are glad to invite and welcome Dr. Serladan Kamalakannan Sir, Professor, Department of Biotechnology from South Korea, as the speaker of this session. We heartfully invite Dr. Pariyapon for sharing this session. And we welcome Dr. J. Sendil Kumar Sir from Department of Biotechnology for joining the session as the co-chair. And we humbly request Sendil Kumar Sir to kindly welcome the speaker and to please introduce him.
Sir? Yes. Sir is working as a full-time professor at University of Jeonbuk National University, South Korea. And Sir has started his career as the Junior Research Fellow in University of Madras and a Research Fellow at Madras during the year of 2002 to 2006. After that, Sir has been working in various designations at the same university, Jeonbuk National University, South Korea. And right now he is holding the position of full-time professor and taking care of the entire department of the same university. I'm happy to say that Sir has been worked as a visiting professor in PSG College of Arts and Science during the year of February 2018 to 2019. And he has been completed his PhD in applied microbiology, applied geology, and interdisciplinary degree, along with his master's in microbiology and bachelor's in general microbiology. To add feathers on his cap, Sir has various awards like 15 overseas and Indian recognitions for his noble work. And he is being a member of various other institutions and agencies like and he is a recipient of Honorable Prime Minister of India, an award which is named as Vaibhav Summit Inauguration Function. And he is an eminent of his biography, Who is Who in the World, right? A book. And he is awarded with postdoctoral fellowships and various other recognition. His field of specialization is metal microbe interaction, plant microbe interactions, and few other biological agents and its controls. And he has published various other research articles in 25 peer-reviewed journals, along with the SCI index papers as 40. And Sir has guided 2 research scholars, and currently he is guiding 1 research scholar. And it's my pleasure and my duty to welcome Dr. Serladan Kamalakannan Sir for this session, who came all the way from his South Korea. I welcome you, sir, and thank you.
Thank you, sir. Now, we kindly request our speaker to proceed with the session.
Very good morning to one and all. Before my presentation, I would like to thank the organizing members and the management and the faculty members of Life Sciences for this wonderful opportunity to share about the research works in my laboratory. The title of my presentation, it is about bioelectrodegradation of xenobiotics. Bioelectrodegradation, it is nothing but we have integrated the conventional biodegradation process with the electrocatalysis, or a conventional electrodegradation process. And let me start with some small introduction about xenobiotics, and then I will share the results what we got in my lab. And before that, I'm very happy to share the information. The part of this work was done by one of my PhD student, and he have completed his master's in PSGCARE, Environmental Science Department. And actually he joined in my group almost 5 years before, And 2 years before, he completed his PhD program, and presently he's doing his postdoctoral fellow in some other university. And then a small introduction about the xenobiotics, especially BTX compounds and bisphenol A. BTX, the acronym stands for benzene, toluene, ethylbenzene, and xylene. So these are the common volatile organic compounds which are present in the hydrocarbons. And hope you may know that hydrocarbon pollution in the ecosystem, it is a major problem in almost all the countries throughout the world. And the major sources for hydrocarbon pollution, so it includes the accidents during oil exploration, and then accidents during refining, storage, and the application of hydrocarbons in industries. So these are the major sources for environmental contamination. And once if the environment is contaminated with the BTX compounds, it increases the chances for human exposure. And once if the humans are exposed with the BTX compound, it affects the function of liver, kidney, lungs, and even the function of bone marrows. A lot of studies correlated the relation between the anemic condition and benzene exposure in the human system. And US Environmental Protection Agency, EPA, they enlisted BTX compound as a top 100 priority pollutant. So it is very important to remediate BTX contaminated soil and wastewater. And then a small introduction to BPA. It is nothing but bisphenol A, and chemically it is called 2,2-bis-4-hydroxyphenyl propane, and it is one of the essential raw material for the manufacturing of polycarbonates and epoxy resins. Again, polycarbonates and epoxy resins, it is commonly used for the manufacturing of several products which we are using everyday life, like water bottles, and then some kind of trays which are used for packing the food materials, meats, fruits, vegetables, bakery products, and even it is used for the manufacturing of water pipes. So all these things, it increases the chances for BPA exposure. And one of the study reported that the BPA concentration in toddlers, it is very high when compared with the teenagers and adults. And in addition to that, the wastewaters from polycarbonate manufacturing units, it is another major source for BPA contamination in the ecosystem. And once we are exposed with the BPA, it affects the function of reproductive system, and it decreases the immune level, it affects the function of thyroid, and it affects the function of liver and some other organs. So again, it is very important to remediate, or it is very important to degrade the BPA from the contaminated soils and wastewaters. So these are the common methods which are used for the treatment of contaminated soils and wastewaters, like a photocatalysis, biological treatment, electrocatalysis, coagulation, etc. And in our case, we have integrated the biological treatment with electrocatalysis, and we say it as a bioelectrodegradation. We integrated the conventional biodegradation process with the electrocatalysis. And the bioremediation or biodegradation, it is collectively called it as a bioremediation, and I hope you may know about it. Nothing but the biological systems are used to degrade the pollutants present in the contaminated soil or water. Whereas the electrocatalysis, so it is nothing but a process involving oxidation and reduction, and it is mediated through electrons. And a small introduction, not a small introduction, the advantages and the disadvantages of biological method and the electrocatalysis method. So the advantages of the biological method, it is one of the eco-friendly method. It do not develop any secondary wastes, and it do not need a high amount of energy. And the most important thing, the biological methods, it do not cause any negative impact to the indigenous biotic communities. Or I can say that it do not have any negative impact to the organisms which are present in the contaminated soil or the native organisms. And the disadvantages, actually the biological method, it takes a long time. So the incubation time for a biological method, it is very long. It is a time-consuming process. For example, so in one of the study, it is reported that these two metabolites, the two metabolites which are present in the BPA contaminated wastewater, even after 1 month of incubation. It clearly indicates that the bacterial system, it needs more than 1 month of time for complete degradation of BPA compounds. And several studies reported that for a complete degradation of BTX compound, the microbial system needs 12 days or more than 12 days. So it is one of the major disadvantages in the bioremediation. And some other disadvantages like limited microbial growth and limited bioavailability. It depends upon the type of pollutant, and it depends upon the environmental conditions like biotic and abiotic conditions of contaminated soil and water. And then advantages of electrocatalysis. So these are the advantages. It is carried out at ambient temperature and pressure, and it is possible to treat wide range of industrial wastewater by this method. And the presence of salts, it do not have any major impact in the electrocatalysis, or it do not have any major impact in the remediation rate. Whereas the disadvantages, high chance for the development of halogenated byproducts, and then fouling of electrodes. And the major disadvantage is this electrocatalysis method, it has a limited application for solid matrix, or I can say that it has a limited application for the treatment of contaminated soils. So to overcome the disadvantages in the electrocatalysis and bioremediation, we integrated the conventional biological biodegradation and electrocatalysis, and then we can say it as a bioelectrodegradation system. And this is the schematic representation for bioelectrodegradation system. So the system, it consists of the reactor, and the reactor consists of 2 electrodes, and the electrodes are connected with the external power supply. And if electric current is applied, the anode, it releases the electrons. The electrons, it oxidizes the water molecules into hydrogen and oxygen. So it creates an oxygen-rich environment around the anode. And this oxygen-rich environment, it attracts the microorganisms. So the microbial population is very high around the electrodes. Again, the high population of microorganisms, it induces the oxidation reduction reactions around the electrode. And the protons, it move towards the cathode, and the protons reduces the oxygen into hydroxy radicals. Again, this hydroxy radicals, it induces oxidation of primary pollutant as well as the metabolites which are produced during the time of degradation of primary pollutants. So this is the mechanism in bioelectrodegradation system. So in addition to microbial activity, the oxidation induced by hydroxy radicals or reactive oxygen species produced in the bioelectrodegradation system, it enhances the degradation rate of the pollutant. And these are the advantages of bioelectrodegradation system. And these are the protocols which we have followed in our lab. So we have isolated the bacteria from contaminated soil or a sludge. And then the bacteria are screened with the help of minimal salt medium supplemented with bisphenol A or a BTX compound as a primary carbon source. And then the potential bacteria is identified by 16S rRNA sequences. And then the metabolites which are produced during the time of bioelectrodegradation as well as a biodegradation. It is identified with the help of GCFID and HPLC MSMS. And then we optimize the experimental variables to enhance the degradation rate. And also we did real-time quantitative PCR experiments to understand the expression of important genes or expression rate of important genes, or I can say that the genes which are involved in the degradation of BTX compounds and bisphenol A. And these are the results for BTX compounds. So we did a preliminary study. So actually we have isolated 4 different bacteria from the contaminated soil. And among this, one of the bacteria, which was designated as YNS1, it was selected for the bioelectrodegradation system because the strain, it exhibited a maximum degradation in the preliminary study. So we selected this strain for bioelectrodegradation system, and this strain was identified as Pseudomonas foetida based on the 16S rRNA sequences. And then we optimized the experimental variables for the bioelectrodegradation system, like we optimized the initial concentration of BTX compounds and then a pH and applied potential. And in the case of pH, we got a good efficiency at the neutral pH. And in the case of applied potential, we got a good degradation at 0.8 volt. And again, initial concentration, the degradation rate was limited or decreased according to the initial concentration of BTX compounds. And so this is the results. So under optimized condition, we got the 92% of BTX degradation within 6 days by bioelectrodegradation system, whereas in a conventional method, the degradation rate is only 54%. Whereas in electrocatalysis, it is about only 10.3%. And these are the reasons for the variations in the degradation rate at acidic pH or alkaline pH or a low applied potential, like enzyme stability and the solubility of BTX compounds and the limited growth of the bacteria. And again, electron shuttle generated by the bacteria at a volt of about 0.8 volt, it is responsible for the enhanced degradation. And this is the picture of the bioelectrodegradation system, a small scale reactor, which we did most of the experiments in this reactor. And this one is the biodegradation rate. So a rapid degradation was observed during the first 3 days, and after that, the degradation rate was gradually increased and it reached the equilibrium at 6th day. And later we did a GFP tagging. So actually we did the GFP tagging to assess the survival of the bacteria in the bioelectrodegradation system. GFP tagging, it is nothing but green fluorescent protein tagging. So we did this by 4 parental mating. Like we have incubated our bacteria or isolate with 3 other E. coli strains. And one of the E. coli strains, it contains GFP2 gene. And the rest of the E. coli cells, it has a helper plasmids. So we incubated all this bacteria. And after overnight incubation, we have inoculated the bacteria in Pseudomonas isolation agar, supplemented with antibiotics. And after incubation, the presence of GFP in our isolate, it is confirmed by PCR studies. And in the top, you can see the picture. So the top one is the wild type strain, and the second one is the GFP tagged strain. The GFP tagged strain, it emits green fluorescence under UV light exposure. So after GFP tagging, we used the strain for bioelectrodegradation system. And during the time of incubation, we have collected the soil samples at 1st day and the 5th day, and we estimated the microbial population. And the results, it clearly indicate that the isolate YNS1, it has a potential to survive in the bioelectrodegradation system. And then we have elucidated the biodegradation pathway. So actually during the time of incubation, we have collected the samples at regular time interval, and then the intermediates which are present in the samples, it is identified with the help of GCFID, and then we elucidated the pathway with the help of KEEG database. And so based on these results, we found that even after 7 days of incubation, the metabolites are not completely degraded in the contaminated soil, and even we haven't found acetaldehyde. Whereas in the case of bioelectrodegradation, we found acetaldehyde at the 6th day, and it is completely degraded into carbon dioxide and water at the 7th day. And these are the enzymes which are involved in the degradation, especially in the bioelectrodegradation, like a different type of monoxygenase gene and different type of dioxygenase gene, hydrolase, hydratase, aldolase. So different enzymes were involved in the degradation. And later we assessed the efficiency of BTX degradation in the presence of, before that, it is a comparison between bioelectrodegradation pathway and biodegradation pathway. So the results indicate that the metabolites which are produced in the bioelectrodegradation, it is completely different from conventional biodegradation. So it indicates that the hydroxy radicals or reactive oxygen species which are produced in the bioelectrodegradation, it enhances the oxidation rate of primary pollutant, or it enhances the oxidation rate of metabolites, and it could be responsible for a rapid degradation. And also, the enhanced oxidation of metabolites, it increases the bioavailability of metabolites to the bacterial system, and it may alter the expression rate of some important genes in the bacterial system, and again, it could be responsible for the rapid degradation. So to confirm this, again, we did some experiment, real-time PCR experiment, real-time quantitative PCR experiment, and we found that there is a difference in the gene expression rate between conventional biodegradation process and bioelectrodegradation process. So before that, so actually we assessed the efficiency in the presence of chromium, BTX biodegradation efficiency in the presence of chromium, and we found that the presence of chromium, it do not have any impact on the BTX degradation, because chromium, it is one of the common metal, it is present in the hydrocarbons. And then, next, okay. So actually we have located some monoxygenase gene, dioxygenase gene, and nitrilase gene, hydrolase gene in our isolate. So this is the size of the gene, and this one is the sequence identity. So after locating this gene, after locating these genes, after locating this gene, we have cultivated the bacteria in a bioelectrodegradation system, and then we have extracted the RNA from the bacteria, and cDNA was synthesized, and the cDNA was used as a template for real-time PCR experiments. And in the real-time PCR experiments, in the real-time PCR experiments, we found these differences. Like in bioelectrodegradation system, the expression rate of all these genes were increased. For example, if you take a nitrilase enzyme, the expression rate of nitrilase gene, it is 7 times higher than the conventional biodegradation process. And similarly, if you take a catechol 1,2-dioxygenase gene, it is about 5 times higher than the conventional biodegradation process. So the results clearly indicate that, so not only the reactive oxygen species, the enhanced expression rate of some important genes, it is responsible for the rapid degradation of pollutant in bioelectrodegradation system. And these are the results for a BPA. So we have isolated the potential bacteria from contaminated sludge, and based on the, we got 8 different strains from the contaminated sludge, and we have selected HV3, it is one of the bacteria, because this bacteria, it exhibit a good growth in minimal media, supplemented with the BPA as a carbon source. And the bacteria was identified as a bacillus species based on the 16S rRNA sequences. And these are the results. Under optimized condition, the bacteria degraded almost 98% of BPA within 6 days of incubation, whereas around 60% degradation in conventional biodegradation and 11% degradation in electrocatalytic degradation. And this is the MIC, and the bacteria can able to tolerate up to 150 milligram per liter of BPA. And then again, we optimize the experimental variables to enhance the degradation rate. And so these are the optimum conditions. And as I mentioned, under optimized condition, nearly 98% of BPA was degraded in the bioelectrodegradation system. And again, limited degradation of BPA in acidic pH or alkaline pH, and high initial concentration and limited applied potential. It could be the following reasons, like limited solubility of BPA and limited bioavailability of BPA and deprotonation of BPA at alkaline pH, and more energetic electrons are produced at 1 volt. So these are the reasons for the variations in the degradation rate of BPA in bioelectrodegradation system. And this one is a morphology of the electrode in bioelectrodegradation system. So actually we observe the morphology of the electrode with the help of scanning electron microscope. And before bioelectrodegradation, the surface morphology of the electrode is very smooth and it is not occupied by any bacterial system. Whereas after bioelectrodegradation, it is completely colonized by the bacteria. it clearly indicates that the oxygen-rich environment around the electrode, it attracts the microorganisms or a bacterial system, so the microorganisms colonized on the surface of the electrode. And here is the toxicity assessment for the remediated soil, BES remediated soil. So if you take the toxicity, so the germination rate, it is good when compared with the contaminated soil, whereas the root length and the shoot length, again, it is better than the contaminated soil. Even the wet weight of the plant, it is better than the contaminated soil. But still, the root length, shoot length, and the wet weight, it is lower than the control soil. It indicates that the remediated soil, it needs some time for the stabilization. And then we have elucidated the biodegradation pathway for bisphenol A, like same thing for BTX compounds. So during the time of incubation, we have collected the samples at regular time interval, and then the metabolites which are produced during the time of degradation, it is identified with the help of HPLC MS. And so initially we observed the hydroxylation of methyl group and hydrolysis. It may lead to the formation of hydroxyquinone, and then a CC bond cleavage in isopropyl rings and the phenol rings. It may lead to the formation of acetaldehyde, and it is completely degraded into carbon dioxide and water. And this biological degradation pathway indicates that the bacteria need at least minimum of 10 days for complete degradation of BPA. But in the case of bioelectrodegradation system, so it is completely degraded in the 6th day. So again, the same process we have observed, like hydroxylation of quaternary carbon and a CC bond cleavage in the aromatic ring. So these are responsible for the degradation of BPA compounds in bioelectrodegradation system. And then again, so we have located some genes in the Bacillus species, lacase gene and monoxygenase gene. And this is the size of the gene and the sequence identity. And presently we are doing some experiments, real-time PCR experiments, to understand the differences in the gene expression rate. And also, in Bacillus species, 4 different pathways were involved in the BPA degradation. So presently we are doing complete genome sequencing for our isolate to understand the pathways which are involved in the BPA degradation. And presently we are doing some experiments to understand the efficiency of the strain for unautoclaved soil, and also the expression rate of the important genes in unautoclaved soil. And also we are planning to scale up the process to understand the efficiency of bioelectroremediation in large scale. And then a conclusion. So this in our experience, we found that the bioelectrodegradation, it is one of the rapid method, and it is one of the more efficient method when compared with the conventional biodegradation process and the electrocatalysis process. And nearly 98% of BPA and nearly 92% of BTX compounds, it is rapidly degraded in the bioelectrodegradation system when compared with the conventional biodegradation or electrocatalysis. And it is one of the inexpensive method, and it is one of the eco-friendly method. And it is very easy to integrate the method with some other conventional methods. And thank you for your attention. And I would like to acknowledge National Research Foundation of Korea for the funding support. So if you have any questions, I'm sorry, because of limited time availability, I did my presentation at very fast. I don't know how much it was useful for you. So still, if you have any queries related bioelectrodegradation, you can contact me at any time. And my email ID is kannan@jbnu.ac.kr.
Any queries by the students and participants?
Thank you very much, Dr. Kannan, for a very informative and really nice talk.
Thank you.
Any questions from the floor, please? Yes. Yeah? And yeah, microphone, please. Yes. You can. This one.
Hello, sir. It's a very wonderful presentation. Actually, it inspired us a lot because we are also working in the same path as you. We are working on biodegradation process.
Okay.
And we are looking for some integrated approaches as well. So when you talked about integration of electrolysis and biodegradation process, we had some few more queries and doubts.
Yeah, sure.
Yeah, so first question is, so you said you will be coating organisms on electrodes. So what will be the procedure of that? Will you be dipping the electrodes into the broth of the microbial culture or?
No, actually, first you need to check the stability of the strain in the presence of the stability of the strain to ability of the strain to survive in presence of reactive oxygen species.
Okay.
And you cannot screen it in a liquid media. So if you want to screen it in a liquid media, you need to place some proton barrier. The concept is almost similar to that of a microbial fuel cells, but in our case we didn't keep any membrane in the reactor because we did the experiment in solid matrix. So if you want to do the experiment in liquid matrix, it is very important to keep the membrane in between the electrodes. Otherwise, the electrodes, it may get fouled and you may not get a good result. So first you screen the isolate or the stability, ability of the isolate to survive in the presence of reactive oxygen species. So we did a preliminary screening like that, and we assessed the potential of the strain to survive in applied potential, various applied potential like 0.2 volt, 0.4 volt, 0.6, 0.8 and some other thing. And based on that, we have optimized for the experiments.
The next question is, as we are using an electrochemical cell, there will be some heat energy that will be generated, no sir?
No, it will not generate. Actually, we used a graphene electrode.
Okay.
The voltage is very low.
Right.
So it doesn't make any impact.
Yeah, so because you mentioned some list of enzymes, so we were concerned whether the heat energy will affect the process.
Heat will not produce.
Okay.
No.
And the next question is, so is it possible to recoat the electrodes with the microbes again and again, or you will be doing some decontamination
Yeah, it is possible
process?
to reuse the electrode.
Okay. No.
Yeah.
If the electrodes is coated with organisms once, can we recoat it, or you have to decontaminate it and recoat it again in order to avoid contamination process?
Yeah, actually we have not coated the electrode with the bacterial system. Right. So we just placed the electrode, and after an incubation of about 6 days, we have removed the electrode, we absorbed it in the microscope. So it indicates that the bacteria are colonized on the surface of the electrode. So as I mentioned, the electrodes, it releases the electrons. It makes an oxygen-rich environment around the electrode. So this oxygen-rich environment, it attracts the microorganisms around the electrode. So the microorganisms, it may get colonized on the surface of the electrode. And I think it is not a big issue. So you can...
Similar to biofilm formation?
Yes, almost similar to biofilm formation. So after some time, you can remove the microorganisms with the help of alcohol or any other thing. And you can reuse the electrode for some other purpose, like for remediating some other pollutant or for using it for another bacterial system. Yeah, it is not an issue.
One more question.
Yeah, sure.
So what about the electrolytes that you're using, sir, in regard with this combination of biological and the electrolytic part?
Sorry.
Electrolytes.
We didn't use any electrolytes.
Okay. Right, okay.
Yeah.
Thank you.
Yeah.
Yes, some more question? Yes.
Sir, I would like to put a question that you told BPA and BTEX reducing organism will take 1 month and 10 days
Yeah.
Subsequently.
Yeah.
Is there any possibility to make the organism with the days reduced instead of 1 month? Can we make it as 10 days? Is it any possibility?
Yeah, I think it is not possible, sir. Maybe if we optimize the conditions, and I think it depends on the bacterial system and the type of pathways which are involved in the biodegradation. So as I mentioned, if you take a Bacillus species, it has 4 different pathways for BPA degradation. But if you take some other bacteria like a Pseudomonas, it has 1 or 2 different pathways. So it depends upon the number of pathways and the type of pathways and the type of the bacterial system.
Okay, maybe genetic engineering approach can be used.
Yeah,
Possible.
but in the case of GMO, the rate of success is limited.
Okay, sir. Thank you.
Yeah, thank you very much.
Yeah, just one question for me.
Yeah.
In terms of application, I mean, of course, for the bioelectrodegradable, is more effective in comparison with the conventional method.
Yeah.
And how about for the cost to operate?
Yeah,
Is it worth to operate by using this technique?
Yeah, it is one of the inexpensive method. It doesn't need much amount of cost. So just the electrodes cost when compared with the conventional biodegradation. So it is one of the inexpensive method, not that much expensive.
Yeah. Okay, that's good.
Yeah.
It's really promising for the industry, right? If we
Yes.
can apply
Yeah, for
some
the
technique
treatment, yes.
that can control the cost.
Yes.
Okay, thank you very much. Ladies and gentlemen, could you join me in thanking Dr. Kannan? Thank you.
Thank you.
Thank you, sir. Thank you, ma'am. On behalf of our organizing committee, we request Dr. Pariyapon to honor the speaker with a memento. We request the chairperson and the co-chairperson to greet the speaker with a pleasantry. We express our heartfelt thanks to the speaker, chairperson, and co-chairperson for engaging in this technical session too. We right now bid for a tea, and we request all the delegates and participants to settle for the next technical session 3 by 12:05. A small reminder for all the oral and poster presenters, those who would like to display your work and present it, please go to the respective venue, and when the technical session starts, please join back for the session.
Good morning. I have great pleasure in introducing my long-time associate, Dr. Shankar Ganesh. He's a great researcher. Sorry, had done his post-graduation at Pondicherry University. After that, he had moved on to the very reputed institute in France and Europe, INSA Toulouse. Toulouse is very near to the France, Paris, and all that. And he has done his PDF there, and he had moved to the BITS Pilani Hyderabad campus. You know the BITS, very reputed BITS Pilani institutions, in which he has been working till now. And he had done a lot of administrative capacity, has done a lot of administrative work as associate dean for the internship and all that, summer school, everything. Moreover that, he is a very good expert in the national level in the anaerobic digestion. He has patented the technology and he has developed the products. In fact, the lot of food waste that's generated in the hostels, all the hostels of the BITS Pilani, I have seen in person when I visited the campus, where all the food waste every day are made into lots and lots of biogas, and that have been used for the cooking itself. So they're recycling it very efficiently. The plant is running very well, and he recently has been promoted as the professor of biological engineering department, and he has got many awards, many projects. To just sum up the recent one, he has got the 25 crore DBT INSA PERS, DST PERS, 25 crore project for him alone. So we have such an eminent research person, experienced the national level, international level, and he has been the speaker for many international conferences, and he is a frequent visitor of our department, not only as our college, as not only the researcher, but also as an administrative person in the NAC and all that administrative work. So he is a very frequent visitor at PSGCARE. He is a good old friend of our institution. I have great pleasure in introducing and inviting him for this great lecture. I ask the students to be attentive and interact with the speaker. He is highly practical oriented, product developing, patent developing scientist. So I request all the participants, the students, the research scholars, everyone to listen to his lecture and interact with him for all the products that he has developed, the rich experience of research and administration, both he has got. So I would like to expect at least 5 to 10 questions after his talk. Thank you so much. Now the dais is Dr. Shankar Ganesh.
We request Dr. Victor Arogya Das sir to present a pleasantry to the speaker.
Yeah. Last verse.
Thank you, sir. Now we kindly request our speaker to please take over the session.
Thank you, Professor Victor. It is good afternoon now. I know this is like a challenging session before the lunch, so I won't take much of your time. At the outset, I thank the organizers for giving me this opportunity once again to be in this institute and then share my experience. I work on a similar field to Professor Kannan. I work on waste management, especially on wastewater treatment through biomethanation. So when I was doing my PhD, many of my colleagues used to ask me, so what was your thesis on? I say it is waste. So every thesis is a waste only, man. What is your actual title? Then I say it's really on waste. So that's how it started almost like 30 years now. I've never deviated from that, and I keep working on waste management. Today I'm going to talk about a specific thing. I'm sure you know about the landfills. How many of you know the landfills? Raise your hand. Okay, good. So this would be the contents next. So today I'm going to talk about a very stinking problem, that too before the lunch session, which is on the garbage, right? So technically it's called as the municipal solid waste, what we all generate, you and me, in the cities. If you see, especially in a country like us, the solid waste gets dumped in the landfills or the dump yards. So there is a black and white difference between the dump yard and the landfill. So dump yard is just like a open area where we just go and throw it. And landfill is an engineered structure where the pollution is limited and steps are taken care for the liquid, the air pollution, and then the groundwater pollution to be restricted. So just to give some figures, globally around 2 billion tons in 2016, and 3.4 is the expected quantum of waste by 2050. And USA alone in 2018, it was 0.3 billion. Next. And in India, 2020, we generated 550 million tons. And this is only on paper. I'm sure what is actually generated and what is being recorded, there will be a lot of difference. And 2021, it increased to 584, at least close to 6% increase in a year. And today we are in 2023, mid of it. So year on year, the waste generation is going to increase. Next. And if you see what happens to the waste, what we generate, almost 50% is being composted. Composted, I'm sure you guys know, so it's just a aerobic biodegradation. And then whatever is being produced as a product of composting can be used as a soil amendment. I carefully use the word soil amendment, not a fertilizer, because that has limited fertilizing capacity. And then if you see rest of it, around 18% goes for landfilling, and almost 32% it's not been accounted. That's why we see a lot of waste on our streets, right? Next. And if you see the global treatment methods for the municipal solid waste, it's almost the same. Around 33% goes to open dump. So no engineered structure, just find a place and then start dumping the waste. And 8% goes to the sanitary landfill. And very less, like 4% goes to the controlled landfill where it is secured and there is minimal chance of pollution to the environment. And 25% landfill but unspecified, whether it is open dump or it's not been controlled, we don't know. 5% for composting, incineration, especially in the European country where they need the heat to heat up warm up the buildings. So incineration is one of the ways. And then 13% recycling and others. This is the global scenario. So whatever it is, you see the open dump, sanitary landfill, controlled landfill, and landfill. All these four contribute towards the landfilling or the dumping of this waste. So all these activities generate what is called as the landfill leachate. So landfill leachate. So this is like a very hazardous liquid which comes out of it. So what you see on the top, this is like an open dump. I'm sure this is a very common scenario across all the cities, especially in India and any other Southeast Asian country, versus what you see at the bottom, which is a proper landfill. So it is covered so that the percolation of the rainwater doesn't happen and then it doesn't sort of pollute the groundwater. Next. So leachate is the liquid which is hazardous in nature because whatever is present in the waste, especially the moisture content, and because of the rainfall, it leaches through the moisture, the municipal solid waste, and it is generated. That's why it's called as leachate. So it is very, very heterogeneous, especially country like us, because we don't separate the waste at source. So we mix the waste and then we throw it in the dustbin and it goes to the landfill. So it will have a lot of other dangerous materials, whatever you name, it will be there. So it's going to be very heterogeneous and it has to be treated. That's the point. So if you see the major characteristics of the landfill leachate, again, we can divide the landfill into two types. One is the active or young, another one is the closed or the old landfill. So young landfill in the sense that it's still in operation, and the closed one, as it says, it is closed. Probably it would have just got filled up, so they close it. So the landfill leachate is generated from both the landfills. Either it is closed or open, and then it has got very weird characteristics, as you could see: high chemical oxygen demand, biological oxygen demand, inorganic compounds, heavy metals. And then when you talk about the closed landfill leachate, it contains something which is very specific, which is the ammonical nitrogen. And in addition to this, there is an organic substance called as the humic substance. Next. So these are additional characteristics of the landfill leachate. So the moral of the story is this cannot be let out just into the environment. This has to be treated, and it has to brought to the set norms by the regulatory bodies before it gets into the environment. But unfortunately, what happens is most of the MSW is being generated in the open dumps. So there is no treatment happening there, and the landfill either goes and pollutes the surface water body or it pollutes the groundwater body. Right. Next. So one of the process by which we can treat any organic matter, including the landfill leachate, because it contains a lot of organic substance, is the anaerobic digestion, which is of these 4 phases. I don't want to get into the details, but what we get out of this anaerobic digestion finally is the biogas. I'm sure you would have heard about this gobar gas, right? Anybody heard about this gobar gas, biogas? Right? So we use the cow dung and we maintain the anaerobic conditions, and as a product, the biogas is produced. Biogas contains, what is the predominant gas in biogas? Methane. So that has got good calorific value, and it can be used as an energy supplement, either directly for thermophilic applications, or it can be converted to electricity and then used for the regular electric applications. So these are the 4 processes. Next. Next. So again, this can be done in 2 different phases, single phase or multi-phase. So this explains how the substrate which is present in any of the waste, including the landfill leachate, can be finally converted to methane and carbon dioxide. But there is a problem here. As I said, in addition to other things which are seen in the landfill leachate, we have these heavy metals, ammonical nitrogen, and then refractory organics. So refractory, the prefix is for those organic material which defy the biodegradation process. So I just had a chat with Dr. Kannan. So he has come up with the technique where he can degrade those things which normally defy the regular biodegradation. So we plan to work together and then subject this landfill leachate to the electrolysis before it being subjected to the biodegradation. So, but as such, if you see, these are the 3 main things which are present in the landfill leachate, which will defy the anaerobic digestion, or for that matter, any biodegradation process. So we need to have some activities so that the degradation can be enabled. Next one. And I want to focus specifically on ammonical nitrogen today. So ammonical nitrogen is usually present as a total, or it is in the form of free ammonical nitrogen. And as we all know, the free ammonical nitrogen is much more toxic than the total, because it can enter into the cell and then alter the intracellular pH. And normally the ammonical nitrogen concentrations more than around 1350 mg per liter will affect any biodegradation process, especially the anaerobic digestion. So I'm sure this is a little bit of chemistry here, but I'm sure you guys should be knowing about the normal nitrogen degradation. So typically in the environment, nitrogen is present in the form of ammonia, as you could see here. And what is the source of ammonia in domestic sewage? Anybody? What is the source of nitrogen in domestic sewage? Sewage, domestic wastewater. Where does the nitrogen come from? No idea? Yeah? Nitrogen? Do we have nitrogen in the domestic sewage? Yes. So what is the source? The excreta, right? The urine especially. But the urea gets converted to ammonia and stays there for a longer period of time. So that has to be treated. So first it gets converted to nitrite, and then from nitrite it gets converted to nitrate. And finally the nitrate gets converted to the nitrogen gas, dinitrogen gas, and it goes back to the atmosphere. So that's the conventional cycle of nitrification and denitrification. So the first process of ammonia to nitrate, nitrite, and nitrite to nitrate, that together is called as the nitrification. And then finally, when it gets converted to nitrogen gas, that's called as denitrification. That's we all know. Next. So just to have a schematic representation, as you could see, ammonia to nitrite, predominantly by the ammonia nitrifiers in the presence of oxygen, and nitrite to nitrate, nitrite oxidizing bacteria, and nitrite to dinitrogen gas by the denitrifying bacteria. So here, oxygen is required in both the steps, that is from ammonia to nitrite and nitrite to nitrate. But nitrate to dinitrogen gas doesn't need oxygen. That's the crux. You need to understand this because I'm going to talk about it in the next slides. So that's the actual requirement. Next. Next one. But we have a process called as the simultaneous partial nitrification, NMOX, and denitrification. This is abbreviated as SNAD. A very promising technique in wastewater treatment, especially for the nitrogen removal. As you could see here, the ammoniacal nitrogen is converted to nitrite through partial nitrification. And then the nitrite is used as the final electron acceptor to oxidize the dinitrogen gas in the NMOX process. So what is that final electron acceptor? Anybody? So normally when you talk about the aerobic cellular respiration, so we have final electron acceptor, yes, the oxygen, right? So here, oxygen, in addition to that, nitrite is used as the final electron acceptor. And then the residual nitrates, they also get converted to nitrites during the dissimilatory nitrate reduction nitrite, which is called as the DNRN process, that will utilize the refractory organics. These are the organic substances which are present, and then it will be converted to nitrite. So next slide. So this is the NMOX process where the ammonia is converted to nitrite, and then one species, which is the nitrate itself, is not being formed. So from nitrite, directly it gets converted to nitrogen gas. So what is the advantage of this? Anybody? What are the advantages of this conversion? So one species is not being formed, nitrate is not being formed. Can you guess? You saw the conventional process, right? So what is the difference? Go to the next slide, I think it is there. Yes, between these two. Yeah, somebody said. Yes, there is a reduction of oxygen requirement in terms of time. So the oxygen, because oxygen has to be pumped in, so it saves a lot of cost. You know, the process becomes economic. Next. So this is like a gist of the SNAD process. Ammoniacal nitrogen will be converted to nitrite, and that nitrite will be again converted to dinitrogen gas. And if little bit of nitrate is present, that also will be converted to nitrite, and again it be converted to dinitrogen gas. So this is a very promising technology which is available for the nitrogen treatment in wastewater treatment field. So in addition to that, there are also indigenous microorganisms which are very important. So when I started my talk, I said that it's very difficult to degrade the landfill leachate because the organisms may not survive in the hazardous nature of the landfill leachate. But if you go to the landfill sites where our garbage is being dumped, there are a few microorganisms which grow in this landfill leachate. So if you are able to identify the microorganisms from the source, which means that they can survive there, we can try to exploit them for our own wastewater treatment process. So it's very important to find out the indigenous microorganisms which can sort of resist the toxicity which is being produced by the landfill leachate. So it is very difficult to culture them in laboratory because these guys live in syntrophic relation. That means the product of one organism becomes the substrate for the other organism. So you can't isolate one organism and grow. It might grow well, but when you put in process, probably it will not work because they work in groups, right? So it's better to go and identify from the site and then try to replicate the same environment in the lab and use it in the wastewater treatment process. So next slide. So this is the methodology what we have followed. So we collected the landfill leachate from a closed landfill because that is rich in ammoniacal nitrogen and then the humic substances. And we fed it into the SNAD reactor. We also have isolated some of the inoculum sources, including the landfill leachate and other things like the activated sludge process, which stands for ASP, And then the lake sediment, just go to a natural environment and take some sample from there and test whether they are good in treating the landfill leachate. And MBBR is yet another wastewater treatment process, the moving bed biofilm reactor. And then we tested the treated effluent for various parameters as well as for toxicity. And we also took out the sludge from the SNAD reactor and did the 16S metagenome sequencing just to find out what type of organisms are there and how the microbial shift has happened. And conclude on the efficiency of the SNAD reactor and possible microorganisms which can help in the degradation of humic substances and the ammonical nitrogen. So, next. So what we could see is that, next slide. So what we could see is that almost 96% of ammonical nitrogen reduction happened in the SNAD reactor. That's a great thing because if that is controlled, then the leachate can be treated by any conventional aerobic treatment or anaerobic treatment methods which are available, including the ASP or the MBBR or the anaerobic digestion, whatever we just saw a few minutes back. And the nitrate was not detectable at all, and nitrate concentrations were lower than 10 mg per liter, which is sort of okay for any type of biodegradation process. And when you talk about the humic substances reduction, so I'm sure you guys would have heard about this word called as the humus, right? Where do you find humus? Pardon? Yes, top layer of the soil. So top layer of the soil. So how is the humus formed? So the leaf litter falls, and then the microorganisms work on it, and then the humus is formed, it gets converted to inorganic form, again the plant takes in, again the leaf litter falls, it goes on a cycle, right? If you see in the forest floor, the top layer especially contains rich of humus substance. So these substances are also formed in the landfill because the landfill, once we start dumping the waste and close it, it becomes sort of a anaerobic condition, and then the organic matter gets converted to the humus form, and further degradation would be very, very difficult. That is why the leachate, when it is leaching from the waste, it also carries these humus substances. But we could see that almost close to 60% of this humus substance reduction was observed through the SNAD process alone. So if you are going to have one more process followed by the SNAD, then we will be able to reduce the humic substances as decided by the regulatory bodies. So with very less DO concentration, 1 to 1.5 mg per liter, we were able to run the SNAD process successfully. Next one. And if you see the COD reduction, the COD, BOD are the very commonly used parameters to study wastewater. People who work on wastewater or from environment, you should be knowing it. So we were able to reduce more than 60% of the COD and to a larger extent the BOD also. If you see here, the BOD reduction is better than the COD because the landfill leachate contains not only the organic substances, it contains a lot of other materials which are present there, which will be very difficult for the biodegradation to happen. And we also did a toxicological risk assessment using the following organisms, and we could find out the risk quotient value got reduced almost to a tune of 94.5%. But anything which is less than 3 only can be considered as it will not cause any risk. So which means that we cannot stop here. So this SNAD treated landfill leachate also has to be treated by one more system so that it doesn't cause any toxicity. Next. We also did the germination studies with Lepidium sativum, and we could see it is not having that much of toxicity. So 75% and 50% of the landfill leachate, when it is mixed with the water which is used for irrigating the seeds, it was producing good germination, which means that it can still be used for the agricultural purpose. And finally, the microbial diversity which is seen in the landfill leachate, which is very important. So we use the 16S metagenomic analysis. And this is the picture of what we got when it was before treatment, right? So you can see some of the bacteria which are dominant there. Next. Next one. So here, next slide. Yeah, so I want to focus on this. So we have the bacteroids and protobacteria. These were involved in the ammonical nitrogen reduction. And we saw that these bacteria, they were abundant in the inoculum, especially those we got from the landfill leachate site itself. And then the proteobacteria, the planktomycetes, so these organisms which are involved in ammonical nitrogen as well as the refractory organics degradation, they got enhanced after the SNAD treatment. So that's the point. So it's sort of a natural selection which happened in the reactors. As a result, the organisms which are involved in the nitrogen removal and the organic substance removal, they got enabled there. Yeah. And linked with the microorganisms are the enzymatic pathways. So the enzymes which are especially involved in the ammonical nitrogen removal, including the hydroxylamine oxidoreductase, nitrate reductase enzymes, ammonia monooxygenase. So these were all found to be more in their activity after they had been exposed to the SNAD process. Next. So few more enzymes which are involved in the conversion of aromatic organic compounds, they also increase during the pre-treatment in the sense like the SNAD treatment of the landfill leachate. And of course, these are a few more enzymes which are involved in this process. So the point here is the organisms and then the metabolic pathways and the enzymes which are linked with the degradation of ammonical nitrogen and the humic substances, they got increased during the process. Next. So to conclude, so ammonical nitrogen and the humic substances, comparatively for any other aerobics treatment, we also do it with other aerobic and anaerobic treatment. Through the SNAD process, it was found to be higher. And growth of specific microorganisms, which are found in the landfill leachate before treatment and also in the inocula. So we used, as I said, 4 different type of inocula we used. So these were, which were having the capability of removing the ammonical nitrogen and humic substances, they got enhanced. Especially these bacteria like protobacteria, planktomycetes and actinobacteria, they were more abundant in the SNAD effluent than any type of other microorganisms. So if you provide proper operational conditions, the SNAD can be a platform for the growth of the native microorganisms which are already present in the landfill leachate. And the toxicity of the landfill leachate reduced after the treatment, but based on the RQ value, it is sure that the further treatment has to be done before it is left out to the environment. So I'll end with this slide because today we are working on the circular economy because till today we have been doing the linear economy where we take out the raw materials, use them, and then we throw it as waste. So it doesn't go back to the cycle. So as a result, we have end up with a lot of waste on one side and we don't have so much of raw material on the other side. So to bridge this gap, we have to talk about the circular economy. So this way of reusing the waste or converting it into a form which can be used again for the production process or manufacturing process is called as the circular economy. So my lab works on the circular economy of waste and wastewater. Next. So these are some of the publications. Next. Next. So which came out of the landfill leachate. Next. Next. So this is a landmark publication from the Stoughton, the Science of Total Environment. Next. So, I mean, last year when I was here, Sir has introduced this book. I have also edited this book on landfill leachate treatment along with my colleague from SRM University. So I would like to thank the sponsoring agencies for my work, Department of Biotechnology, BIRAC, and the Department of Science and Technology, and also BITS Pilani for enormously contributing towards this research. Next. Thank you. And yeah.
Thank you for the nice presentation,
Yes.
Professor.
Yeah.
It's a time for discussion. So if you have any questions, you can discuss with the professor.
Sir, I will start with my questions first. I have two questions. The one is, you said that the leachate has got a lot of chemicals, and as a biochemist, I may not know much about your digestion and all that, but as a biochemist, I'm very much afraid of aware of the chemicals, the carcinogens. So even if you do all the digestion, everything, composting, but then finally so much of amount of leachate, how would you treat that? How the chemicals are, where are they dumped or where are they cured, and how do you get rid of those chemicals? First question. Second question is regarding the 16S genomic sequencing. Do you find the biodiversity you have shown? So in the consortium, from place to place, from waste material to waste material, is there any kind of diversity or relationship or some relationship? Yeah.
Sure. Thank you for the question. In fact, 15 years back when I started to work on landfill leachate, this was the first question I had. So we know that a lot of chemicals and other inorganic substances are there. How are we dealing with that? If you see the conventional methods, what happened today in India or anywhere, they use a process called as the reverse osmosis. Like what we have it in our homes, right? We have this Kent and water filters and other things. Even for a small unit for 4 people in a home, the annual maintenance contract would be around 5,000-6,000. They come and say the filter kam nahi kar raha hai, which we have to change, etc. But how can we how can we treat so much of landfill leachate which is generated across the globe? But unfortunately today, that is the only method they are using it. And once they pass through the reverse osmosis and whatever comes out, they use it for their own process. And then the solids, these are supposed to be very hazardous sludge. So that is being dumped in another landfill. So there is no complete treatment for the landfill leachate at all. So we have mounds and mounds of these hazardous sludges being dumped across the globe, including in India. I'm sure it should be there in Coimbatore also, right? So especially the carcinogens, they all get screened out through the reverse osmosis process. That's the only thing which has been advised by Government of India today. But that's not going to be sustainable. That is where we are looking for alternate solutions, especially the biological methods, which are much cheaper. We are very specific, and they don't generate such sludge, which will be converted for example, if this sludge is being used for anaerobic digestion, this can be converted to energy. It can be used for thermophilic applications or converted to electricity. That's what we are trying to do. That's the first thing. Second thing, the characteristics of the landfill definitely will depend on so many factors. One on whether it is active landfill or closed landfill, and of course from place to place. For example, we have one next to the campus in Hyderabad. So that the government has made it a point that the organic waste should be composted and only the inorganic waste has to be dumped because we don't have space. So in that Jawaharnagar dump yard facility, it receives close to 8,000 tons of garbage per day. 8,000 tons. And then it is estimated in a city like Hyderabad, the daily garbage production is around 11,000 tons. 11,000, tons, right? 11,000 kilograms. So I think this hall is not sufficient to hold it per day, per day. And where do we dump the waste? We don't have space. So that's one point. So coming to your question, of course, it depends on the type of waste which is being dumped in the landfill. The characteristics do change. Yeah. Any other question?
Good afternoon, sir. First of all, thank you for this wonderful presentation, sir. As nowadays in India, especially, we need such innovative ideas and methods to eradicate or to mitigate some problems like this. Now we have discussed about landfill leachate mitigation problem, and the similar problems can be also dealt with the sewage treatment, sir. Actually, as a part of IIC, Institute of Innovation Council, there are many programs like Smart in India Hackathon and all. In that, most of the ideas the government of India need is sewage treatment thing. So I hope similar ideas can be applied on the sewage treatment plant too, sir. Actually, my question is, I'm a third UG student, biochemistry department. So, what are the PG options in life science for BITS Pilani? So,
Okay. So I will answer the first question. Second question we will discuss offline.
Sure. Okay, sir.
So, see, sewage treatment is much more structured as compared to the leachate treatment. Because even today, we are now slowly moving towards the gated communities, the apartments, etc. So before the government gives the approval for building such structures, communities, it is mandated that they should have sewage treatment plant on site. So that is taken care there. Let's talk about old cities where such a structure is not there. The sewage is being collected and it is expected that the urban local body does the sewage treatment somewhere else. While whether it is doing or not, how much is the efficiency, it's the next question, but there is a structure like that. But landfill, I mean, we don't even know, how many of you know where does the garbage you throw every day morning goes? Right? Very few of us might know. Because every major city will have few dump yards and this will be dumped there. And what would happen to the waste, we don't know. And landfill leachate comes as a consequence of dumping this waste and nobody bothers about it. So way back in 2010, very close to our campus is like, I was in, so Sar knows, so I was working in France before joining BITS. So I find no difference living either in Toulouse or in Hyderabad. But the only curse to the campus is the dump yard. So we get terrible stink from the dump yard if the wind direction changes. So once I was very curious and went to the dump yard and I saw this landfill leachate flowing across the road. From the landfill side, it crosses the road and goes to the other side. And to a periphery of more than 10 kilometers, it has completely spoiled the surface water body. And I was told that the landfill is there for more than 40 years. And you could imagine what would happen to the groundwater. It would have already got polluted now. So that's the situation. Nobody bothers about the landfill. That's what we want to focus on this. Sewage, of course, it's also important, very interesting area, but at least some care is taken to that. But this is like an orphanage, right? So landfills are like orphanages. That's why I want to focus on that.
Actually, we are very grateful for this innovative idea, sir. Thank you so much.
Thank you.
Professor, I have one question.
Yeah.
So, is this method have any impact on other type of pollutants? Because you mentioned that the dioxygenase enzyme and different type of monooxygenase enzymes were involved in this process.
Correct, yeah. That's a very interesting question because for this talk, I have focused on nitrogen. It also has especially impact on the humic substances, which I little bit touched upon. But another important pollutant when you talk about landfill leachate is the presence of high phosphate elements there. So SNAD could also be scaled up to other organic and inorganic substances which are present in the landfill leachate. But these two are the key ingredients which defy the biodegradation process. That's why we wanted to focus on that. But this is part of my PhD student's thesis work who defended last week. He got the award. So there in his thesis, we have done a complete characterization of the landfill leachate and post-treatment, how they change, we have presented it. I just taken one slice of it and presented here, focusing on nitrogen, because these are very critical factors.
Okay, thank you. And any more questions from audience? Okay, thank you, Professor. Thank you for your wonderful presentation. Yeah.
Thank you, sir. Now we request the chairperson to outline the technical session.
Okay, so actually in this session, in this session, the presentation is about the leachate treatment, landfill leachate treatment. So I think it is a wonderful presentation by Professor Shankar Ganesh, and hope you may have a good information, or you may have gathered a good information about the leachate and the problems which are associated with the leachate, and the treatment methods for the leachate. And so I think a professor is an expert in the management of the leachate. So if you have any questions, you can ask him directly or in person. Yeah, thank you.
Thank you, sir. Now, on behalf of the organizing committee, we request the chairperson, Dr. Serladan Kamalakannan, sir, to honor the speaker with a memento. We request Dr. Victor Arogya Das, sir, to join with the chairperson to express our gratitude to the speaker with pleasantry. Thank you, sir. We herein wholeheartedly thank the speaker, chairperson, and the co-chairperson for making their way in this technical session 3.
I have, with the permission of the chair, I have one request and announcement is that we need good PhD students for our research. I'm sure this is a universal problem what we face. Either it is in Korea or in India.
Dedicated students.
Dedicated students.
That is important.
So we expect, at least from pioneer institutes like PSG, sir is an alumnus of this institute. So we expect the people like you to come forward and then join us. We have a lot of opportunities today, but we don't get, unfortunately, I'm sorry to say this in the, I want to register it here, we don't get the dedicated students. Because doing research, somebody has to really to be dedicated. And when I was doing, I used to bring the municipal solid waste, the garbage truck inside the university campus, right? People will get scared why the municipality vehicle is coming inside. So change my dress and then we jump in the field and work. Today my students, I'm so scared to tell them anything about this. So we expect many of you to become successful scholars and then join the research community. It's a different feeling. You will enjoy it once you experience it, right? Thank you. Thank you so much.
I have been visiting SAR's campus for the past 5 years. A wonderful campus, brilliant students, and his lab is so equipped. And recently he's published a very high impact factor ranking paper. What is that ranking impact factor? Science of total environment that he had shown, very high impact factor, and was a very big paper with so much of engineering, environmental engineering coming up, estimations, and it's a big advice for a whole municipality or a big city can deal with that paper. What are the projections that given the paper? Such a very informative and very calculative, estimation of the dump, the recycling, all that. Such a nice paper. I've read it. So they do a very high quality research, and the institution has got a lot of funding, lot of machines and facilities, and real place to work with. So I just support this SAR's information that it's a very good place to go there and aim for. Thank you.
Thank you, sir. That's really motivating, and it's a great opportunity for the student as well. Now, we start with the technical session 4. We humbly invite Dr. C. Kadirvelan, sir, Associate Professor, Tamilnadu Veterinary and Animal Sciences University, Namakkal, to please honor the dais. We also humbly invite Dr. K. Ramesh, General Manager, R&D, Grasm Industries, Aditya Birla Group, Mumbai, to join the session as the chairperson. And we request Dr. Radesh Sri, Associate Professor, to join the session as co-chairperson. We request Dr. Thamre Selvi ma'am to kindly welcome and introduce the speaker of the session.
My warm greetings to all the dignitaries and participants. Dr. C. Kadirvelan, Associate Professor and Head, Department of Animal Nutrition, Veterinary College and Research Institute, Tamilnadu Veterinary and Animal Sciences University, Udumalpet, has completed his undergraduation in veterinary science, post-graduation, and doctorate in animal nutrition. He pursued doctorate in animal nutrition at National Dairy Research Institute, Karnal, Haryana. He has received Fellow of Animal Sciences. His field of expertise is on lab animal nutrition and nutraceutical and functional foods. He has completed more than 10 funded projects and 2 ongoing projects. He has also published more than 90 papers in indexed reputed journals and contributed for book chapters. Ireland, Belgium, Thailand for his knowledge sharing. He is a university gold medalist in PG, and to his credit, he has received several awards for his notable contribution to his field of research. I request Dr. Sir to check the bill.
Thank you. Thank you very much, ma'am, for nice introduction. Respected chair and dear delegates, good afternoon. I'm going to talk about production of designer or functional foods and its health benefits towards a human being. Next. See, today we are having a lot of lifestyle-related diseases, especially diet-related diseases. So diet is one of the important factor which determines the health of human beings. That's why we are giving a lot of importance on diet, especially natural ingredient which act as a medicine. That is a prime area nowadays. We are looking for a part of the food which act as a medicine. So consumers like human beings also, even in pets, we are going for natural nutrient which act as a medicine. Next. So consumer preference towards purchase of food in 2000, first preference on low fat diet, followed by functional and low sugar, organic and fortified diet. Whereas now we are going for towards functional food, then followed by organic. We are not saying organic now, strictly saying it is near organic. 100% organic we are not saying, we are not claiming. It is near organic food. Then also low sugar, low fat and fortified diet. Next. So father of medicine Hippocrates, he given a slogan that let food be your medicine and let your medicine be your food. Next. So what is functional food or nutraceutical? All are same. Sometimes we are using designer foods or health foods or even functional foods, all are synonym. A food or functional food is a part of food which having beyond its nutritional property, it's having some medicinal property. That's called functional food. Next. Actually, the nutraceutical coined by a scientist named Stephen D. Fleece. It is a word from nutrition and pharmaceutical. As similar with functional food, a food or part of food which acts medical benefits beyond its nutritional property. Next. So in this context, I am going to discuss about how to produce designer milk or designer egg and designer meat. I think before lunchtime, I think we will have a virtual food world, right? Okay, next. So what is designer milk? So milk is the complete food which is consumed globally. Even vegetarian, non-vegetarian, everybody having the milk, okay? So we are having a lot of health benefits in the milk. So current topics, we are therapeutic aspects of milk. We are looking forward for therapeutic aspects of milk. Okay, India is the #1 milk production country in the world. Almost 240 metric tons of milk is produced per year. Now we are focusing on towards quality, not quantity. Even though we are producing #1 in the world, we are unable to export because of presence of some antibiotic residue in the milk or aflatoxin. That is a mycotoxin present in the milk. So we are targeting towards the quality, not quantity. Quantity we already achieved through white revolution, so many things, right? So how to modify, how to alter the milk composition? We can modify the protein source, or we can modify the fat content, or we can modify the lactose content. But very fragile and very easy to modify is modification of the fat. That's the need of the hour also. We no need to modify the protein or lactose content, but you can modify the fat content in the milk. Next. So how we can manipulate the milk composition? The milk fat composition depends upon which type of food we are giving to the animals. They're determined by type of food given or offered to the dairy animal. Okay, so especially milk synthesis and milk component only controlled by the feed which is offered to the dairy animal. Especially in the milk fat, there is a component called conjugated linoleic acid. Normally, you know what is linoleic acid? It's a double bond fatty acid, C18:2, whereas it's become conjugated when double bond takes shift in the place, it's become conjugated. So that's called conjugated linoleic acid. In short, we are saying CLA, right? Next. So CLA is a mixture of positional and geometric isomers. Okay, we are having almost 28 positional and geometric isomers. In this, two biologically active isomers, cis9 and trans11, and trans10 and cis12 is more biologically active among 28 isomers. So mainly food from animal origin, especially dairy animals, rich in CLA. That's conjugated linoleic acid. Right. Next. So, as I said, milk from the dairy animal is the rich source of CLA. Normally, the milk contains 5 milligram per gram of milk fat. You know what is the fat of milk? Now, on an average, 3 to 4% fat is there in the milk. So, it is present in the milk fat portion, that is 5 milligram of, per gram of fat. Why this CLA? We are talking about CLA nowadays. Because I did my PhD in National Dairy Research in Karnal in 2007. I'm part of the PhD in Ireland. From, I think, in 2000 onwards, we are talking about the CLA. Because it's having a lot of health benefits towards the human being. Next. So as I said, it is from dairy product. For example, in a homogeneous milk, it is 5.5 mg per gram of fat, like butter fat, cottage cheese, up to buttermilk. And an average ranges from 3.5 to 6 milligram of milk fat, the CLA concentration. Next. So how this biosynthesis takes place in the dairy animal? So as I said, it is determined by the feed which offered to the dairy animals. A feed which contains C18:2 and 18:3 fatty acid, that is linoleic and linoleic acid. It has to go to the rumen. You know, the dairy animals, there is a two type of animals in the veterinary. One is dairy animal which is having 4 compartments in the stomach. Whereas dog, cat, other single stomach, monogastric animal. This is the uniqueness in dairy animal because that's why it's able to digest the fibrous material in dairy animals. So first part called as rumen, where it's having microbial digestion, like bacteria, fungi, and also protozoa, which digest all fungi, all fibrous material. So when the feed which rich in this fatty acid that go biodigestion process and synthesis CLA and get into mammary gland tissue and excreted in the milk. This is a biosynthesis of the CLA, right? Next. So how we can enhance this CLA content in the milk? And an average 5 milligram, I want to enhance that CLA content in the milk. Through diet, I am going to increase the fatty acid. For example, I can add sunflower oil because rich in linoleic acid, almost 67% it's having. Similarly, I can go soybean like that. N number of oil cakes or oil can add in the diets. Especially I did work on mustard oil, which contained almost 41.2% linoleic acid. So it is give you a substrate for conversion of CLA. Right, next. So in buffalo, we did a trial on buffaloes. One group we thought mustard oil, other group we had mustard oil 2%. In control, we had 6.84 milligram of CLA. Whereas when I add mustard oil, it gives almost 19.5 milligram CLA. Right, next. So why? Again, the question, why we are focusing on CLA in the milk? Next. It's having a lot of health benefits activities, start from anti-cancer, anti-atherosclerosis, immunomodulation, even anti-adipogenic property. Next. When work on mammary cancer, the CLA and mammary cancer, it is a chemopreventive property against mammary tumorigenesis. There are a lot of mode of action we are having, like induction of apoptosis, programmed cell death, also inhibit cancer cell proliferation, cell mitosis, also inhibit COX-2 enzymes and arachidonic acid metabolism de-altering. Finally, is also having free radical scavenging property. Next. So in the trial, we want to check whether this really CLA acts as an anti-cancer or not. So for that, we took a rat and we induced a cancer in the rat. Then we fed that milk. For convenient handling, the milk converted as a ghee. Because milk means we need a lot of quantity. But if you go for ghee, it is a totally fat fraction. So easily we can store, easily we can handle. So in that, I include almost 10% of ghee in the test group, whereas control group for fat equivalent, I took soybean oil. Next. So we induced cancer through 7,12 DMBA, dimethylbenzandrogen chemical agent. So it induced mammary cancer. Next. So almost we took 32 weeks of this trial after DMBA injection. So parallelly, both the groups are induced cancer. One group without CLA-ghee, other one with CLA-ghee. Next. This is the incidence. In tumor incidence, the control group almost 83.33% incidence, whereas in CLA-fed group animals, only 46% of incidence. Even if you go for pathology, histopathology, we categorize this tumor category. Fibroma, adenoma, and fibroadenoma, these are benign type of tumors, whereas adenocarcinoma, it is a malignant type of tumor. But that instance was nil in case of CLA-fed group, but it was present in controlled animals. Okay, next. This is the tumor appearance. One is lateral view, other one is ventral view. Next. After existence of tumor, almost 3 centimeter diameter, we found the tumor size. Whereas in CLA-PET group, next, it's almost 1.1 centimeter. That is a benign type of tumor, not malignant type of tumor. Next. So histopathology to confirm that in soybean group, there is the oil-fed group. It is adenocarcinoma, malignant type of tumor. You can see it is a hyperchromic nuclear changes in the memory tissues. Also, it's the anisokaryosis and anisocytosis, different shape, different in the colors of the cells, memory cells, right? Next. So on liver enzymes, regular your antioxidant enzymes. In the CLA-fed group, it enhanced antioxidant activity. Next. Also T-bars, on the target tissue. Here target tissue is the mammary gland. So in T-bars also it is comparatively less when compared to a control group. Next. So next property, again you want to check whether it really acts as anti-atherosclerosis. Next. So in this we took again almost 12 rats we took in a group. 16 weeks we fed that ghee. So we took a baseline value of all lipid profile. After 16 weeks, the CLA-fed group, HDL was increased, whereas LDL was decreased. This is the result we got it. On histopathology, we given a liver. You see, in a soybean group, there's a fatty generation of liver cells, hepatocytes. Okay, so very clearly it was there. Whereas in CLA-PET group, it not there. I was able to see the portal triad. There's three veins are there in the liver histopathology. Next. So immune system. Definitely the CLA act as a immune enhancing capability. So there's different authors reported. Of course, we did a small trial on immune system, especially on mammary gland cancer-induced rat, plasma nitric acid. Next. So this is a plasma nitric acid level. So in CLA-PET group, it was lower when compared to non-CLA-PET group. Next. So what is the practical utility? Okay, we did a trial. We enhanced the CLA in the milk. So what is the difference between non-CLA milk and CLA enhanced designer milk? So this is a normal milk. I told you it is 5 milligram per gram of milk fat. That is normally present in the CLA. So to achieve a target concentration, we need 20 milligram per day. Every day we need 20 milligram of CLA to achieve beneficial action. For that, how much I have to take? I have to take almost 300 to 600 ml of almost gram of milk every day. It's not possible. Even average national milk consumption 330 gram per day. So 600 gram of milk, it is not possible to take. For that, if we enhance the CLA, I can take only 150 gram per day. That's almost 160 ml per day. This is the advantage. So if you design the milk to particular compound, either fatty acid or vitamins, so particular compound can be increased. That's called designer milk. Here I designed the milk with the CLA, conjugate linoleic acid, because this CLA having lot of health beneficial property. Okay, right, next. So next one is your functional designer meat. And being a veterinarian, I had to talk about eggs and broiler meat, because we know a lot of myths are going on whether I have to take broiler meat or eggs, how many eggs I can take per day. So many myths and so many debates are going on in social media. Even last week we conducted an expo at Namakkal Veterinary College. The Poultry Federation of Tamilnadu, they came and gave an awareness program. They distributed free eggs to all the visitors. They distributed free meat to all the visitors. Okay. Even during the corona, this was a big debate whether if you take broiled chicken or not, if you take corona will come. So many debates was there, right? Right. Next. So you know this is the egg production strategy. I mean status of in India as well as world. So we were in 4th place in egg production and 5th place in meat production. Next. We are having different types of egg because just have a look. We are start from quail egg. This is a very smallest egg. Whereas you know that is the last one is ostrich egg. How many have seen the ostrich? Very few, only 2. So please visit our veterinary college, Namakkal. We are having everything. We are giving as a memento in the ostrich egg only, every seminar. Okay. So, so it is a quail egg from ostrich egg. I think Yumu egg, you know, it is Erode is the good place, Yumu scam, right? So, we can manipulate anything in the egg composition through diet. Next. So, we can manipulate with a particular nutrient that's called designer egg or designer meat. Okay. So, we can manipulate either minerals in the egg or vitamins in the egg or omega-3 fatty acid in the egg or pigments. Have you taken or boil? Have you seen the egg yolk color? I will show how we can compare what is the original egg yolk, what is the fake egg yolk. Okay, next. So these are normal nutrient composition. So there is some myth. If you take more egg, you will get a cholesterol, right? So what is the cholesterol content? Almost 230 milligrams mg. But we need more than 230 milligram per day. Moreover, our own body is synthesizing cholesterol. And on average, we can take 2 to 3 eggs. An adult body weight can take 2 to 3 eggs without any harm. No harm at all. Okay? Don't afraid to take eggs. Even above 60, they can take 2 eggs per day. Okay? Next. So, where we can manipulate the egg? One from your omega-3 fatty acid, we can manipulate, we can enrich. Second thing, low cholesterol eggs, because one group in the world they are always targeting on low cholesterol. In the world, always there are one group working for positive means, other group working for negative. Both are research only. Okay, you have to accept both. If you are working for positive aspect, definitely there will be one group working for negative aspect, right? So iodine, because you know now iodine, you know goiter is there common, iodine deficiency in India, very common. So we are having iodine enrichment, yolk pigment color. Okay, that reflects your carotene contents. Then also herbal, nowadays herbal, again that's another area where we can focus on herbal tea, herbal egg, herbal meat, everywhere it's there. Okay, next. So we can fortify or we can manipulate or we can value addition all these nutrients, start from iodine to xanthine, that's a pigment. There is a very lutein also a pigment, okay. So all these vitamins, omega-3 fatty acids, lutein, xanthine, we can manipulate very easily in poultry. Everything through diet, right? Next. So also minerals and vitamins. Now the minerals concept getting changed. We are using previously inorganic minerals. You know inorganic. Now become chelation. Now the organic minerals we are getting in the commercially. So what is the advantage of organic minerals? To have more bioavailability, so more fast absorption in the small intestine. That will be incorporated in the target tissue. Your target is egg or meat. So we are going for towards in organic mineral sources, especially zinc. Okay. Now we are zinc enriched eggs are there. You know what is the role of zinc in the during corona? It is what is zinc? What is the basic property for zinc? Anybody? Why you are taking zinc tablet for during corona? Or even they given the eggs also for hospital patients. They are given eggs, right? What is the basic property of zinc? Anybody? Nutrition, dietitian departments. It's an immune modulator, right? Zinc is the immune modulator. This is the question asked during my IAS interview. I given my IAS interview main. Being a nutritionist, they asked me what is zinc, what is the basic. I given so many answers. But the chairman board, except one thing I didn't tell. That's the reason. There's immune modulator, immune enhancer. Basically, all pediatric syrup you take, they're having zinc to enhance the immune property of children, right? Okay, so we can fortify. There is a difference between enhancement and fortification. But some way, one point, all are synonyms, right? Next. This is a vitamin enrichment. So first one is regular leg. If we add vitamin enrichment strategy, we can get more amount of vitamins in the egg. Next. This is the herbal leg. Again, herbal leg, we are having different opinions. Still debates are going on, whether really it's herbal or like organic, whether really it's organic or not. That's why we are using the term called near organic, not organic. Any products nowadays not a complete 100% organic. It is near organic product, right? So under organic source, we can include a lot of phytocomponents, phytochemicals in the diet that will reflect on the target product, that is egg or meat. Poultry, why we are selecting poultry again? Poultry is a very short generation interval, right? 35 days per broiler. For laying, almost 14 weeks, birds start laying. So that's why genetic improvement is very fast in poultry, because initially a broiler we reared up to 12 weeks to get 2 kg of meat. By genetic improvement, now 35 days we are getting 2 kg of meat. That's why last of rumors are going on. The peoples are giving hormone injection, no. Or some antibiotics using, that also no. Because of genetic improvement, we are getting body weight 2 kg at the age of 35 to 40 days. It's not to hormone, it's not to antibiotic, it's not to any biotics, right? Next. So we are having n number of phytocomponents that can be added in the meat or egg. Next. So this is herbal enriched eggs. It's having different egg components, especially on egg cholesterol and selenium, because selenium is very important. What is selenium basically? What is the property of selenium? What is the property of vitamin E? Vitamin E is an antioxidant. Vitamin E and selenium both are interrelated. So both are act as an antioxidant through glutathione peroxidase enzyme, right? So both are having sparing action. Suppose vitamin E is deficient, sufficient amount of selenium is there, it giving a sparing action of vitamin E, right? Next. So now how to enhance the chicken meat or chicken egg with the vitamin, omega-3 fatty acids. Next. So omega-3 fatty acids mainly your EPA and DHA. The two main omega-3 fatty acids. The source is from oil seeds. Mainly linseed oil is a source for enhancing omega-3 fatty acid in the egg or even meat. So animal source, we have to include the fish meal, silkworm FIFO meal. Both are rich source of this DHA and EPA that can be incorporated in the egg as well as meat. Next. So this is the linseed. Next. Then how to enhance the egg yolk pigment. This is the actual egg yolk color. This is, I think, you can differentiate both, right? That one is the actual egg yolk color. How many are appreciated this yolk color during your kitchen? Anybody appreciated different egg yolk color? So what is the main component to give a color for egg yolk? There is maize. So in field, whenever the maize price goes up, they will replace maize with the broken rice. And if you include the broken rice, the yolk color comes down. So easily we can understand that these eggs mainly fed with the maize component or egg with the rice component, right? Next. Then organic eggs, as I said, it is not 100% organic, it is near organic. So organic means without any antibiotics, antimicrobial, coccidiostat, other drug. There should be no vaccination, no cages mainly. You should be in free-range. Eggs getting from free-range bird without these activities. Next. Then functional meat. Next. Next. Put it next. Next. So in meat, again we are bothered about the fat content, right? How we can modify the fat? One is reduction of fat content or modification of fatty acid profile. In fatty acid profile, we can reduce short chain fatty acids. Then improving unsaturated fatty acid content. Saturated fat has come down and unsaturated fatty acid has to be increased. Next. Then TFA, total fatty acid has to be reduced. Next. Then CLA, of course. In poultry also, we can enhance very mild amount of CLA in the meat. Right? Next. Again, the next one is again cholesterol. Cholesterol can be come down through feeding strategy. Next. So these are omega-3 fatty acids. What is the importance of omega-3 fatty acid in the human being? Mainly it is improve vascular permeability, fetal development, then also Alzheimer's disease. Okay, you know what is Alzheimer's disease, a lot of research is going on, already done, right? Next. So nanotechnology, other one is nanotechnology. Now we are talking about everywhere nanotechnology, nanotechnology. What is actually nanotechnology? Anybody, nanotechnology department? Mainly it is a particle size less than 100 nanometer, right? That is the thing. So how we can enhance nanotechnology in the poultry, either egg or meat? Because I told you, for minerals we are using inorganic to organic, we are shifted. Now we are going towards the nano minerals. So nano selenium and nano zinc. Zinc, you know, I already told you it's immunomodulator. We want to enhance the zinc content in the egg. So for that we are using nano minerals. Next. So these are nano technology application, especially in the poultry as well as general food sector, especially nano selenium and nano chromium. Yes, we are talking nowadays chromium also. Initially it was in under cumulative toxicity. Now chromium become a conditionally essential mineral for human beings. Why it is conditionally essential? What is the role of chromium in the human being? It is basically a glucose tolerance factor. That's why diabetic patient, so we are having chromium coated tablets we are using. Next. Next last category, phytobiotics, that is plant extracts. There is different synonymy we are having, phytogenic feed additive, then phytogenics, plant extracts, or herbal additives. All these a natural growth promoter. I told you just now, we are talking about antibiotics. Nowadays in poultry industry, they are using antibiotics. Due to that, we are having a lot of complications, lot of debates are going on. Now one group working on replacing complete antibiotic in the poultry and using phytobiotics. Next. Next, future topic, now people are working on nutrigenomics. How the nutrient and gene interaction. There is very, very upcoming field. We are working on the this nutrigenomics. How we can target gene through feed. How we can target gene with a nutrient. So we can manipulate the nutrient, manipulate the gene. For example, a fat synthesis in the cow is controlled by a particular gene. We can target that gene, we can manipulate, and we can produce low fat milk in the dairy animals. The next. So we add a project, development and evolution of micronutrient fortified chicken eggs to combat malnutrition. This is a DBT project. Ours the lead partner, Tanvas, then industry partner, Krishna Poultry. So we completed this project. So by this we enhanced we addressed malnutrition, especially we enhanced some of the minerals. Next. So summary of my presentation, very simple, let food be your medicine. For that we have to design our foods, either milk, meat or egg. Thank you.
Thank you, sir, for delivering an interesting session with the concepts on the importance of conjugated fatty acids and strategies of design of foods with therapeutic applications. Now we request the chairperson to brief on this technical session.
So it was a nice presentation. Before switching over to question hour from student or audience side, so I have two questions. One is, what is the technology gaps in CLA? Then next question is, what is free radical oxidation? How we can avoid it?
In technology gap in CLA, actually we had tried up to level of increasement 19.5. We want to increase up to 40. Further, still people are working on how to enhance the CLA content in the milk. Because when you're enhancing CLA content in milk, we can reduce the daily day recommendation. Now it is almost 150 ml. We want to cut down to at least 50 ml. 50 ml milk we can easily afford, or 100 ml. So still group are working on that. Second thing, free radical scavenging. Any antioxidants in the food, either from vegetable or natural antioxidant or synthetic antioxidant. For example, in poultry, we are using synthetic antioxidant to enhance the antioxidant activity of meat or egg. So any accident activity that can scavenge a free radical generated in the body.
Any questions from audience side can interact with the speaker.
Good afternoon, sir. Your topic is so interesting. So I have one doubt. So nowadays there are many articles regarding lab meat. They are constructing meat in lab. So can you say something about this lab meat? Is it nutritionally supporting?
Yes, it's not nutritionally supporting. Anything against natural, please ma'am, we can manipulate against natural some extent. 100% against natural we can't manipulate. We can manipulate some extent, some percentage. This is lab meat, still it's not commercial, it's still in R&D only. People are saying, I told you one group always working towards the health benefits, one group always working towards the other end. Both are research. We take it as research, but it's not authenticated, it's not good also.
First of all, thank you so much for such a wonderful presentation, sir. So I'd like to ask, you'll incorporate phytochemicals in eggs now, sir. So what are the phytochemicals that you'll incorporate in eggs, and what stage will you incorporate those phytochemicals in eggs?
Okay. So we are having a list of phytochemicals, start from very simple, turmeric powder, right? Ashwagandha, turmeric, even tulsi extract. All having their own phytochemical constituent with having some definite action. These phytochemical we can add in the poultry diet. You know, in poultry there is two category. One is for meat purpose, that's called broiler. That we can rear up to 35 days or maximum 5 weeks. Whereas other category is called layer that give eggs. The layer start laying eggs at the age of 16th week. It will continue up to 72 weeks. We can incorporate this phytochemical from 16th week onwards. We can continue up to 72 weeks. The egg is the reflector. What we are giving in the food or feed, that will reflect in the egg composition. Only thing, the conversion efficiency we have to study. Suppose you are giving 10 milligram turmeric, okay? What is the active component in turmeric?
Curcumin.
Yes. So curcumin I am giving 10 milligram in the diet. How it get converted into egg? How much it get converted in the egg? That is the real interest of research. Sometime in the early age, if you start 16 week, the conversion is more, whereas in compared to 72 weeks. Now it is 80% means in early age. In age later on, it is around 40%. There is a different. The conversion is different. We can add any throughout the life. Start from egg and cessation of egg laying. That is stopping of egg laying.
Sir, will those phytochemicals cross-contaminate with the nutrients present in the egg?
No, it won't contaminate. We are selecting phytochemical because phytochemical having some definite active components and definite action, beneficial action. It won't cross-contaminate. Even if combination of phytochemicals we are using.
Okay, sir. Thank you so much.
Thank you, sir. Now, on behalf of the organizing committee, we request the chairperson, Dr. K.
Madam,
Ramesh.
please. Somebody ask.
Sorry.
First of all, good afternoon, sir.
Good afternoon.
Sir, my question is a simple question. The CLA synthesis is based on the feed given to the cattle, as you told. If the intake of feed is high in cattle, whether it will cause any disease or it will enrich the production, sir?
You told this question is simple, but the question is very tough. Okay, so you told animals taking more food, okay, right? Dairy animal taking more amount of food.
Yes, sir.
Right.
Like
Due to that, it's having more amount of secretion of CLA, maybe having any detrimental effect. Right?
Yes, like
okay,
that.
Right, okay. What's your food intake daily?
Sir,
What's your BMI?
sir, I don't know.
What's your height and weight? How to calculate BMI? So there is that idea is everybody having their own BMI, own intake limit, right?
Yes.
Like that, dairy cow can take per day almost 30 kg of green fodder. That is the standard. For us, it is high. For cow, it is low.
Okay, sir.
30 kg of green fodder, 10 kg of paddy straw, Vaikol, 10 kg of paddy straw, 4 kg of concentrate feed. Punak 4 kilo saapla. Adala normal animal. Normal digestion process, it need. Because what is the body weight? Almost 350 kg body weight is the dairy animal. For to satisfy this, it has to take all these things. 4 kg concentrate, 25 kg green fodder, almost 10 kg dry fodder. In this, it will secrete only 5 mg of CLA per gram of milk fat. That's all. Right?
Okay, sir. Thank you, sir.
கை தட்டிட்டாங்க அதனால நீங்க பண்ணிடலாம்.
So nice presentation. Hope students and audience have enjoyed this session. Particularly, speaker has explained the importance of chromium and zinc. It was really impressive. And thank you very much for your presentation.
Thank you, sir. Thank you once again for organizer and PSG, chair and co-chair. Thank you very much. Thank you.
We request Dr. K. Ramesh sir, the chairperson, and Dr. S. Radheshri ma'am, associate professor and head of the Department of Food Processing Technology, to acknowledge the speaker with the memento and the pleasantry. We gratefully thank the speaker, chairperson and the co-chairperson for their support rendered for this technical session. We now request all the delegates to kindly proceed for lunch and to please join for the further technical sessions sharp by 2:15.